Good morning, everyone. Just as a little disclaimer again, I don't have a position in BNGO, so I'm just trying to give you an unbiased view of what's going on with this company by holding these live sessions on their symposium. That way the scientists can speak for themselves and you can decide whether or not you want to invest. So, one thing I did want to bring up before this symposium starts, they did open an option chain for BNGO stock, and that's something that somebody in the chat pointed out. That was so great stock, so thanks for that. And that should be available from what I've seen on Thinkorswim, Webull, but I haven't checked Robinhood yet. So be watching out on the option chain. There's probably a lot of people putting their bids in for it. And since there's not really, since it is just opening, there's going to be a lot of demand for it, which means the implied volatility is going to be very high. So whenever you open your position, you might be paying a little too much just because of the demand for these options since they're brand new. So I just wanted to let you guys know about that. It is available on Robinhood. NKC confirmed. Thank you, MKC. Best mod ever. Anyway, so I am just gonna let the science speak for itself. Just wait for these presenters to come on. It's about 3 minutes out. And if you guys have any questions before this begins, let me know. I'm gonna make this live window bigger just so you guys can actually see. Actually, do they have a full screen version? Yes, they do. Cool. Also, can anybody confirm whether or not they're playing their elevator music right now? Just want to make sure that the audio output is correct this time. Alright, so it's 10 a.m. They should be starting any second now. Of course, Juice, thanks for being here. If you guys could run up the likes on this video, push it out to more people, get this information out there, that'd be much appreciated.
Check.
So MKC just confirmed that there is elevator music playing, so I'm gonna have to try to reroute this audio before they begin. Hopefully we can do this. I don't know why it's always their presentations. Yeah, I might have to refresh the stream. Give me 1 second. Let's see. F11. I don't even know how to exit this. Let's see. There we go. Let me try to refresh. Maybe that'll help. All right. So can you guys hear? I think there's supposed to be audio.
Welcome everyone to Bionano's Next Generation Cytogenomics Symposium. We are on day 3. My name is Alka Chauve and I am the Chief Medical Officer of Bionano Genomics. So just a little bit of a recap of yesterday, where we had an incredible session on hematological malignancies, where you can see the snapshot of our 8 speakers from yesterday. And for those of you following us on social media, please remember hashtag Bionano Symposium. So today, January 13th, our session is focused on solid tumor applications. But please remember, we have 2 more days, January 14th, where we will be focusing on repeat expansions and complex molecular diagnostics. And we have a bonus day on Friday, January 15th, where we will have an expert panel from the COVID-19 Host Genome Structural Variation Consortium. So to remind everybody, all of the speakers for this Next Generation Cytogenomics Symposium are from across the world, where we have the majority of our speakers from the United States, Europe, and then we have some from China and Australia. A few housekeeping notes. Today is day 3 of our Next Generation Cytogenomics Symposium. For those of you that have not registered for Thursday and Friday, please go back to the registration page where you can check these boxes and a link will be sent to you. Please remember, if you have not checked these boxes, a link automatically will not be sent to you. So why are we here today? Why are we talking about structural variation detection? Because for all of the neoplasia subtypes, whether it is hematological malignancies or solid tumors, medical associations from across the world, such as World Health Organization or National Comprehensive Cancer Network and rest of the world, recommend the detection of structural variations as the first-line test for the diagnosis of these neoplastic disorders. If we look at the statistics from World Health Organization, you can see this chart where you can see for both sexes of all ages, the incidence of these solid tumors or breast cancer, prostate, lung, and colon cancer, and so on and so forth from across the world. Yesterday, we had a session on hematological malignancies, including leukemias and lymphomas. And in the inset, you see the prevalence per 100,000 cases. And just to bring this reference to everybody, we see the leukemia incidence highlighted in yellow, which essentially now you can see is dwarfed by the enormous amount of new cases estimated for solid tumors, and also in an enormous amount of deaths are expected to occur from these neoplastic disorders. If we look at the American Cancer Society statistics, in 2021 alone, in the United States, there will be an estimated approximately 2 million new cancer cases and more than 600,000 cancer deaths. This amounts to almost more than 5,000 new cases and more than 1,600 deaths occurring every day. That's pretty phenomenal statistics in terms of prevalence and mortality. So it is really important to understand that for the solid tumors also, these different medical associations have recommendations and guidelines, such as WHO and NCCN, that recommend cytogenetic testing, either using karyotyping and FISH, or using molecular technologies such as microarray or other methods such as MLPA, and also having molecular genetic testing for targeted sequencing variants. And this is a common theme for as part of recommendations for diagnostic workup and testing. Again, why is cytogenomics important in neoplastic disorders? If we go through the literature, the reason is that there are unique structural variants that are associated with each tumor subtype. And these structural variants include translocations, fusions, inversions, copy number alterations, and chromothripsis or other genomic instability events occurring throughout the genome. On the right-hand side, we see this table from NCBI, which talks about these somatic events associated with a specific tumor subtype. I must emphasize this list is not complete. If you start going through each cancer subtype, there are an enormous amount of aberrations that are prognostic and diagnostic, which we will see today our speakers talking about their research and their clinical evaluation. So structural variants can be among the hardest to detect genomic variants. If we look at sequencing technologies, which are used for identifying or detecting single nucleotide variants at base pair resolution. Also, we have conventional methodologies that are able to identify aberrations in full chromosomes, such as aneuploidies. Then we have a range in the middle, ranging from hundreds of base pairs to millions of base pairs, where structural variations are actually dependent on the limit of detection of each technology to identify the accuracy of these structural variants. This is a slide courtesy of one of our collaborators, Dr. Eddie Maher from the United Kingdom, where he put this together to demonstrate that in a conventional cytogenetic laboratory and in a diagnostic workflow, there are a number of technologies that are used to come to a diagnosis, which include karyotyping, microarray, and FISH. So not only is this entire framework slow, but it is also extremely complex, as you can see from the slide. On the right-hand side, you see the Bionano Saphyr workflow for optical genome mapping, where you just have a very simple workflow to get to the diagnosis. Now, sequencing technologies have been reported to perform poorly for structural variants, especially short-read sequencing really have poor performance for large structural variants. We have several long-read sequencing technologies which are not cost-effective and have inconsistent throughput. On the other hand, optical genome mapping using Bionano's technology has the right throughput, is cost-effective, and we can have as low as 5% allele fraction at 400x depth and can have as low as 1% allelic fraction detection at 1600x depth. So let me introduce optical genome mapping with our Saphyr platform. On the left-hand side is this video, live video of these long molecules of DNA that are imaged in our Saphyr instrument, which is the basis of the optical genome mapping technology. So when the labeled DNA is loaded onto the Saphyr chip using electrophoresis, these DNA molecules are untangled and then linearized in these nanochannels, which are on our Saphyr chip. On the bottom right-hand side, you see an actual image of these molecules, these ultra-long molecules, where the backbone is labeled in blue, and the green dots represent the unique, specific labels across the genome. So for solid tumor, right now, we have a very simple and single workflow where we can start with approximately 5 milligram to 50 milligram of tissue as the starting material. We have special DNA isolation protocols to isolate ultra-long DNA molecules, which are then labeled uniquely at a 6-nucleotide motif across the entire genome using a special enzyme. Then these molecules are transferred onto our Saphyr chip. And if you remember, you can appreciate the video that I showed you on the previous slide, which is showing how these molecules are shooting through the nanochannels and then imaged onto the instrument. After these high throughput, high resolution, ultra long DNA molecules are imaged, then they are aligned to a reference genome to identify structural variation patterns. So structural variants by optical genome mapping are detected by comparing the patterns of the labels to a reference. So in the top, you see in green is our reference genome pattern of all the labels. And in the blue are our structural variation maps for a given sample of interest. So a deletion would result due to either a loss of labels or a decrease in distance between two labels. And an insertion similarly would result either due to an increase in distance between two labels or the introduction of additional labels. Also, repeat array expansions can be accurately assessed looking at the pattern and the number of repeats of those labels. And a duplication event can easily, just based on the pattern recognition, be inferred to be either in tandem or reverse orientation. But one of the key advantages of using optical genome mapping is the ability to identify translocation events occurring either between two or multiple chromosomes, where just based on the label patterns, we can identify the translocation breakpoint and which chromosomes are taking part in this event. An inversion can also be identified just looking at the inverted pattern of these specific labels. Along with the ability to look at these structural variation maps, there is also a unique algorithm that allows us to get the copy number variation profile from chromosomes 1 through 22, X and Y, which a lot of the viewers might be used to because this is very similar to how a microarray whole genome profile is. So structural variation classes. If we look at aneuploidies, we have the ability to pick up monosomies and trisomies. And for copy number variations, for deletions, we can pick up interstitial or terminal events. And for duplications, it is very easy to identify tandem, inverted, or insertions occurring on other parts of the genome. Similarly, insertions of either known or unknown sequence can be detected, and translocations, whether they are balanced, unbalanced, or cryptic, can also be easily identified. The inversion patterns to correlate to whether it is a pericentric or a paracentric event can also be detected along with other abnormalities such as ring chromosomes and other repeat contractions or expansions. We do not detect single nucleotide variants. So I want to orient all the viewers to what they will be seeing in our list of speakers today and when they are presenting their talks. So all the optical genome mapping data will be presented in this circus plot, which the outer ring shows the ideogram of the entire genome from chromosomes 1 through 22, X and Y. And then starting from the outside, the next ring shows all the different structural variants with different dots. So you can just look at the different colored dots to see whether it is an insertion, deletion, inversion, duplication, or translocation. This is followed by a track, which gives us the copy number variation profile. And then the inner ring gives any translocation event occurring between different chromosomes or other events. So in this example, there is a translocation event occurring between chromosomes 11 and 22. Also, the software allows us to filter all of these structural variants against our population control database, and users can use their own custom vet files to analyze a specific tumor of interest. So please remember this when you look at all the data presented today in our solid tumor session. We have an amazing speaker lineup today, which will be followed by our panel discussion and live Q&A. Please do not forget to stay till the end and join all of these speakers and me for our live Q&A. We are going to start with Dr. Jim Broach from Penn State Cancer Institute, where he will be showing his work on HPV integration correlating with genomic instability in head and neck cancer. This is followed by Dr. Adrian Lee from UPMC, where he will show his cases of how he used optical genome mapping to resolve complex rearrangements in breast cancer. Then we will have Dr. Justin Balko from Vanderbilt University, where he will show his work on identifying large structural variations in non-inflammatory and inflammatory breast cancer. And then we will have Dr. Eric Letouze from Cordelier Research Center on his work on using optical genome mapping for unraveling complex structural rearrangements induced by replication stress in liver cancer. We will have Dr. David Chen-Tien from Institute Curie, who will then talk about how he is using optical genome mapping to identify different events in aggressive uveal melanoma models. And then we will have Dr. Ravindra Kole from Augusta University, who will show us his work on the clinical evaluation using optical genome mapping for the assessment of genomic aberrations in solid tumors. Please remember that we will have a live Q&A and join me and the speakers at the end of this session. Thank you.
Hello, it's a pleasure to be here to talk about work we've been doing over the last couple of years on the nature of the association of the human papillomavirus in head and neck cancer. So most cancers over the last several years have been on the decline. One of the few exceptions to that has been oropharyngeal squamous cell carcinoma, oropharyngeal cancer, which has been rising since the middle of the 1980s and is projected to continue to increase in the near future. This has been associated with an increase, a dramatic increase in the percentage of cancers, oropharyngeal squamous cell carcinomas, in which HPV has been found. So in the mid-'80s, only about 16% of oropharyngeal cancers carried human papillomavirus, whereas as recently as 2004, over 70% of the cancers are HPV positive, and that number continues to increase. So we know that human papillomavirus has been associated with cancers, particularly with cervical cancer. And from work on cervical cancer, we know a lot about how HPV might interact with the genome and its role that it might play in driving tumorigenesis. In cervical cancer, HPV initially infects the epithelial cells and resides as an extra chromosomal element, as an episome, basically, replicating independently of the chromosomes. As the cancer progresses, HPV becomes integrated, and during that integration, one of the viral genes is often lost, in this case, the E2 viral gene. That leads to increased expression of two viral-encoded oncogenes, the E6 and E7 oncogenes, which respectively inactivate RB and P53. RB is a cell cycle inhibitor, so that its inactivation leads to uncontrolled cell cycle, and E7 inactivates P53, the guardian of the genome, so that the loss of P53 often leads to increased genome instability, which further drives the cancer. So that's what's known about HPV as it was analyzed in cervical cancer. And studies to date suggest that the story is similar in oropharyngeal squamous cell carcinoma, but not identical. And one of the difficulties is actually understanding and being able to determine in a particular cancer and a particular tumor whether the human papillomavirus is integrated or remains episomal. So we have been using the new optical mapping technology developed by Bionano in order to address that question, not only to understand the state of the HPV in the human genome in these tumors, but also the state of the human genome in those tumors as well. So we've been applying optical genome mapping or whole genome imaging to a number of oropharyngeal squamous cell carcinomas, made feasible by the recent development that we worked with investigators at Bionano to develop, that is the ability to extract high molecular DNA from solid tumors. This has made it possible to treat solid tumors, much in the way we've been able to analyze liquid tumors such as leukemias, by being able to identify almost the entire karyotype of these solid tumors. So we've applied that technology again, as I said, to squamous cell carcinomas of the oropharyngeal, and combined that with whole genome sequencing. So we merged the data we generate from the optical mapping and the whole genome sequencing to identify all of the structural variants as well as single nucleotide polymorphisms that are present in those tumors. At the same time, we do the same analysis on blood samples taken from those same patients. And so we can identify in the tumor data those structural variants or single nucleotide polymorphisms that are polymorphisms and retain only those that are somatic mutations. So in applying those to a number of tumors, we've found some very intriguing information that I'll take you through now. So our first patient was a 67-year-old male with a growing neck mass, which is evident in this MRI over here on the right. You can see on the left side of the neck this large tumor. On excision and biopsy of both the tumor and the lymph nodes, it was clear that this tumor was P16 positive, which is a good indication that it's HPV positive. The whole genome sequencing, the whole genome imaging revealed the structural variants that are shown in the circles plot, which I'm sure you've seen in many of the talks presented previously. In the circles plot, the chromosomes are arranged in a clockwise fashion from chromosome 1 around to the X chromosomes. On the inner portion of the circle are the translocations and inversions. In the next ring are is indicated the copy number of those chromosomes. And then in the middle ring are shown the insertions, deletions, duplications, et cetera. So as you can see in this tumor, there are a number of translocations and inversions, as well as a number of insertions and deletions. From the whole genome sequencing, we were able to identify both human and tumor reads, and in addition to identify actually reads in which the tumor sequences were adjacent to the viral sequences, which is an indication that the HPV genome is integrated in those cases. And the tumor sequences, the human sequences that were adjacent to the HPV genome sequences mapped to chromosome 15. And if we look then at the corresponding region on the obtained by optical mapping, we see at this point an insertion and duplication, which when we, as shown here, and then if we drill down on that from the optical mapping data, the site of integration, we see the structure on the bottom here with 2 copies of HPV inserted in a tandem duplication of the surrounding genome sequences, which lead us to suspect that the order of events that led to this structure in the tumor was integration of the HPV genome, followed by mobilization and duplication of the 20 kb region surrounding that insertion, leading to the tandem duplication of the virus and its surrounding sequences. In the second patient that we looked at, a 61-year-old male, we found the patient presented with a large mass at the base of the tongue, which is evident over on the right in this PET scan. Again, on excision and biopsy, the tumor was identified as being P16 positive, indicating it's likely HPV positive. In this case, though, the whole genome sequences failed to yield any hybrid human viral sequences, and the optical mapping showed no insertions or duplications within the genome, nor any other structural variants for that matter. We've now been able to apply this to a dozen samples, 11 of which are shown here. And from the whole genome sequencing, we were able to map the sequences of the viral genome that were present in these tumors, and also identify whether those tumors had HPV integrated into the genome or whether the viral genome remained extrachromosomal. These ones in violet are tumors that were extrachromosomal, in which the viral genome remained extrachromosomal. And these ones identified in teal are tumors in which the HPV virus genome was integrated. And in addition to just getting copy number, we could also identify point mutations or variants within the HPV genome. And what is evident here is that the HPV genomes that were present as episomes were basically didn't constitute a distinct strain from those in which the HPV was integrated. so that the integrated versus episomal state of the virus is not a consequence of the virus per se, but is results from some other extraneous event. And looking then, much as we did in the first tumor, at the structure of the genome around the site of integration, we see that in every case in which the virus is integrated, there are significant structural rearrangements associated with the integration. So this is the event that I showed you initially, where the virus integrates, and then the 20 kb of human genome around the virus is amplified and duplicated. In a second tumor, we have a similar event in which the virus is integrated. The 90 kb region around the site of integration is mobilized and triplicated in a tandem orientation. In the third case, the virus is actually integrated as a consequence of a 3-strand translocation event in which the virus sits at the junction in a translocation between chromosome 11 and chromosome 8, so that the resulting chromosome is an 8-11 chimera with the virus at the center of that translocation. But at the same time, there was a reciprocal 8-11 translocation, as well as a large deletion of sequences on chromosome 11. In the fourth case, we have integration of the virus and mobilization of the 30 kb, in this case leading to 5 copies of the integrated virus and surrounding sequences, as well as duplication of an adjacent region of the genome. We also found in half of the cases that the virus integrates not at a single site, but at 2 sites on separate chromosomes in the genome. In this case, the virus integrates both on chromosome 20 and on chromosome 11 with a complex duplication, again, as an intrachromosomal translocation, with the virus sitting at the junction of the head to tail duplication of those host genome sequences. Another case of the virus integrating at 2 different sites, again, a complicated situation in which the virus is integrating at 2 separate sites, one on chromosome 8, one on chromosome 19. Again, the virus integrating at an intrachromosomal translocation at the junction of the head to tail. And then in this final case, a very complex situation in which, again, the virus is integrated at the junction of an intrachromosomal translocation with the head to tail duplication, and some of the intervening sequences duplicated in a very complex expansion of about 2.5 megabases of sequence with 26 different copies of the virus. I should point out that simply this structure was determined directly from analysis of the optical mapping data, so that the optical mapping gave us a very clear indication of the events that were happening at the site of integration. But the more striking aspect of these is the difference in the whole genome structural variation in the episomal versus the integrated cases. On the top here, on the top row, are the Circos plots of the genomes of those tumors in which the virus remains episomal. And as you can see, there are absolutely no translocations or inversions in these genomes, and few, if any, insertions or deletions in the genome. On the other hand, in every case in which the virus is integrated, there are a large number of translocations and inversions, as well as a large number of other structural variants, including insertions, deletions, and duplications. Just looking at the numbers, these on the left are are the the numbers of insertions, deletion, inversions, duplication, and translocations, and the cases of episomal tumors, whereas these on the right are the numbers found in the tumors in which the virus remains episomal. As you can see, there are absolutely no translocations or inversions in the episomal tumors, whereas in all of the cases of integration, there are a large number of integrations. This structural instability also extends to a certain extent for somatic point mutations. Those samples boxed here are the ones that are episomal, whereas the others have integrated HPV. What is shown is the number of somatic point mutations of various types in each of the tumors. And as is evident, those tumors with episomal HPV have significantly less point mutations than those with integrated HPV. So this raises the question of cause and effect. That is, does HPV integration induce genome instability, perhaps as a consequence of increased E7 expression leading to reduced p53 levels, or perhaps to the focal amplification leading to genome-wide instability? On the other hand, it's possible that genome instability actually results in HPV integration, that perhaps some global genome catastrophe generates a number of double-strand breaks that capture the episomal viral genomes that are resident in those tumors. So we don't know the answer to that yet, but our data to date is more consistent with this latter interpretation, basically based on the fact that we see the cases in which the virus is integrated at multiple sites within the genome, which is much more consistent with a concerted event following a genome catastrophe rather than a sequential events associated with selection for increased viral expression. So what is the consequence of the integration versus episomal state? These are the outcomes data or the characteristics of the tumors. And while we don't have enough data to draw statistically significant events, these data suggest that the tumors that are associated with integrated HPV are more aggressive than those associated with the episomal HPV. In particular, if we look at the tumor staging, the tumors with integrated HPV have a higher tumor staging than those with episomal HPV. Similarly and relatedly, the mean tumor size of the tumor and the metastases are larger for tumors that have integrated HPV than they are with episomal HPV. So in conclusion, we can conclude that we have validated a new technique to utilize optical genomic mapping to construct the genomes of solid tumors. Optical mapping is able to construct complicated genome alterations that were previously unidentified using whole genome sequencing or even more complicated protocols alone, such as the focal amplifications induced by viral genomes. The optical mapping is a powerful tool for identifying and characterizing viral genomes in tumor. From our data, from our limited data to date, we can see that HPV integration is strictly associated with significant genome instability. And our data suggests that that integration is associated with more aggressive tumors. So we think that being able to identify HPV status in these tumors may in fact provide a prognostic and a therapeutic tool for clinical investigators. So with that, I'd like to acknowledge the folks that did the work, those in the Institute for Personalized Medicine and those in the Department of Otolaryngology. So thank you very much for your attention.
Welcome. I'm Adrian Lee. I'm the professor at the University of Pittsburgh, and I'll be talking today about optical mapping to resolve complex DNA rearrangements in breast cancer. These are my disclosures. None of them are relevant to the presentation I'll be giving today. So my lab is interested in understanding the genomics of breast cancer, and through the Cancer Genome Atlas, we know quite a lot now about the genomics of breast cancer and other cancers. This is from Nature Genetics 2013, where the TCGA group separated cancers into whether they are the M class, i.e. they are dominated by base pair mutations, or they're the C class, they're dominated by copy number aberrations. And as you can see here, in the M class, you have kidney cancer, colorectal cancer, glioblastoma, lung. But my interest is in breast cancer. And you can see here, this is actually dominated in the C class by copy number aberrations, similar to ovarian cancer. There aren't that many base pair mutations. And these copy number aberrations and DNA structural aberrations have been inherently much harder to detect than these M-class base pair mutations. So here's an example of the complexity that we see in breast cancer. This is genome rearrangements in the MCF7 breast cancer cell. This is the most commonly used cell line in breast cancer. And as you can see, this is spectral karyotyping, and it shows that this cell line is near triploid, and there are translocations pretty much in nearly every single chromosome. You can see all these different colors. And this highlights the tremendous complexity of the genomic rearrangements across this cell line. I was fortunate enough to be with a group that was the first to develop methods to do this by next generation sequencing. So this was an early mate pair sequencing technique that gave base pair resolution of all of these structural rearrangements. And you can see this here. This is a circus plot with chromosomes around the outside. You can see these interchromosomal rearrangements, and every chromosome is rearranged. And it's amazing this cell line can actually grow in culture given the complexity of how the DNA is intertwined in this cell line. Now, many have actually then translated these DNA structural rearrangements into understanding the biology of breast cancer. And I just highlight one paper that actually came out this year where they looked at DNA copy number motifs, i.e. how each of those DNA copy number structures look, and found that they were strong and independent predictors of survival in breast cancer. So here's just an example from one of the figures, 3 different types of rearrangements they see. And these 3 types of rearrangements have been reported before. This one has been called simplex because you see here, there aren't many DNA copy number rearrangements in this genome of this breast cancer. Here you have complex, where you have now these rearrangements that copy number amplifications, deletions that seem to be clustered on certain chromosomes. And here you see what's called sawtooth, where basically, like that MCF7 breast cancer cell line I just showed you, they have copy number rearrangements across the whole of the genome. This tends to be the most aggressive cancers, like triple negative breast cancer, and this tends to be the more non-aggressive, like the luminal estrogen receptor positive breast cancers. Importantly, these different types of rearrangements have prognostic importance. So they calculated a classifier for these different types of rearrangements, scored those, and you can see there are some that have low risk when you have this score and have good outcome breast cancers, and then there are some that then have high score and then high risk. This is done in 2 different large data sets. Now, not only do these structural rearrangements have prognostic significance, they also potentially have therapeutic value. We know some of them already have therapeutic value. The HER2 amplification, which is one of the determinants of breast cancer, one of the major features, is obviously therapeutically targetable with anti-HER2 antibodies such as Herceptin. we have been performing structural rearrangement analysis in breast cancer and found other potentially therapeutically important structural rearrangements. And here's an example. This is a primary breast cancer where we did mate pair sequencing. You can see this is from an estrogen receptor positive luminal breast cancer. Not many structural rearrangements in this breast cancer. But when this recurred in the lymph node, this is about 5 years later, you can see now there are many more structural rearrangements. And importantly, one of them here, from chromosome 5 to chromosome 6, is actually in the estrogen receptor itself, one of our main targets in estrogen receptor positive breast cancer. What happens in this patient at this specific location is the estrogen receptor, which is here, structurally represented here, gets cleaved halfway down and gets fused to this gene called DAB2, which at the time we found this, not much was known about this gene. And you end up with this fusion gene here, half of the estrogen receptor and half of this DAB2 protein. This creates, as the title suggests, a hyperactive ligand-independent estrogen receptor, and I show you this here. This is a reporter activity for estrogen receptor activity. When you have wild-type normal estrogen receptor, estrogen increases the activity. Anti-estrogens, which are important in the treatment of breast cancer, block that. So this is a classic response. This is that fusion from this patient. You can see, first off, it's hyperactive. It's much more active, 25 times more active than regular estrogen receptor. And then when you give hormone, it's completely ligand independent. So this patient was being treated with hormone therapy, yet there was obviously going to be no response because the version of estrogen receptor they have, this fusion, is completely unresponsive to hormone. And probably this played a dominant role in the progression and unfortunate death of this patient. These, as I said right at the start, these are very hard to detect, these fusions. The technologies to detect them are still challenging, but we've now detected many of them and others. The red ones are from our group, the black ones from others' group. And you can see these estrogen receptor fusions are recurrent, particularly in endocrine-resistant, advanced, and metastatic breast cancers. So I'll show you what we've done now moving towards another form of breast cancer and how we've used optical mapping to try and understand a unique subtype of breast cancer. So this is invasive lobular breast cancer, ILC. It represents a subtype. About 10 to 15% of all invasive breast cancers are invasive lobular breast cancer. If considered an independent cancer alone, it's the 6th most common cancer type of women, with about 26 to 39 new diagnoses a year in the US. And what we show here is the unique features, unique clinical features, unique metastatic features, and unique biology of this invasive lobular cancer, this unique subtype. Its pathognomonic feature is mutation of E-cadherin. So about 58% of ILC have a biallelic mutations in CDH1, generally a loss of heterozygosity and a point mutation on the other allele, causing loss. And the other forms of ILC have some other methylation and other forms for loss of E-cadherin. So nearly all of them have loss of E-cadherin, generally through mutation. So despite this being a very unique genetic form or a unique genomics of breast cancer, it's treated nearly identical to invasive ductal cancer, which is the major form. And there isn't really much research into this form of breast cancer, particularly because there are very few models to actually study it in the laboratory. And our lab has a strong focus trying to understand this. So as I said, the unique feature of ILC is the mutation and loss of E-cadherin, which is present in nearly all ILCs. There are only 8 published cell lines for use, of which only 2 to 3 are actually publicly available. And only 2 of these have been characterized by DepMap. There are, however, additional ILC-like cell lines that phenocopy ILC, but actually aren't called ILC. They're annotated as invasive ductal cancer, or IDC. And we believe there are critical need for additional ILC models and have many studies underway to do this. And I'll explain how we're using Bionano optical mapping for that. So here's an example of these ILC-like cell lines. So this is the ILC cell lines. As I told you, there are 8 of them. This is the ILC-like cell lines that phenocopy the ILC. And this is an example of how we show that phenocopying. So this is E-cadherin here in MCF7 cells, that cell line I showed you right at the start. That's an invasive ductal cancer cell line. It has E-cadherin. The cells have adherence junctions and bind tightly together because of the E-cadherin. This is an ILC cell line, MM134, right here. It lacks E-cadherin, that's why it's ILC. And because of that, the cells now don't attach to each other. And you can see there are individual cells here because of the lack of E-cadherin. And that's the phenocopy and genotypic arrangement for ILC. This is a cell line we made from an organoid, from an ILC patient. You can see it lacks E-cadherin and the cells look the same. But this cell line, ZR75-30, which is here, is a cell line which you can get from ATCC. It's labeled as IDC, yet you can see it lacks E-cadherin and it phenocopies basically ILC. And we're very interested to understand, well, are these cells actually invasive ductal cancer, IDC, or are they actually genetically ILC? So what we're doing is we've taken all these cell lines and we're doing DNA analysis, RNA analysis, protein analysis, and other. And I'll show you what we've done with optical mapping, which already has told us some very unique features of these and exciting features of these ILC-like cell lines. So this is optical mapping for the first 9 cell lines. You can see, just like the MCF7s, they all have highly rearranged genomes. They all have unique features. Some of them have very few rearrangements, as the MM134 cell line, and some of them have pretty much the whole genome rearranged, just like the MCF7 cell line. And you can start already to see patterns of different arrangements in these cells that may relate to their aggressiveness or other kind of phenotypic features, which we're trying to relate to now. When we look at copy number data, you can see the copy number data for SNP array, which we already had on these cell lines, looks nearly identical to the data we've now got from the optical mapping. So this is an average of the copy number across all the cell lines. And you can see from SNP array data and Bionano, it looks nearly identical. So it's good that Bionano data has given us something that looks very similar to tried and tested technologies. We find major DNA structural rearrangements called chromothripsis. So this is looking at chromosome 17 here, and then chromosome 11 down here. And you can see through these mapping that optical mapping gets you, you can see lots of these blocks of DNA have been rearranged together. And obviously the genes within those have been totally scrambled and totally rearranged. And this is an example here where this gene here and this gene here have been turned around, reduplicated, put back together again. And you've basically scrambled not only the genome, but the transcriptome, and then ultimately the proteins that are getting made with different fusions and different gene expression. So what happens in these ILC-like cell lines when you do this to E-cadherin? This is the key question. So this looks at the copy number status for E-cadherin or CDH1 in the ILC cell lines and the ILC-like. You can see there's a number that have copy number loss in the ILC-like. And you can now start to see very unique features of the CDH1 locus in those what are ILC-like, labeled as IDC, but we think they're ILC-like cell lines. So this is one, HCC2218. You can buy this from ATCC. It's labeled as an IDC cell line. And when we get optical mapping data on the CDH1 locus in that cell line, it's very interesting. It has copy number loss on one allele, and on the other allele, it has a very specific deletion right around the CDH1 gene. So you see this singular gene deletion here that's given to you and very clearly resolved by this Bionano map. So ultimately, this cell line has basically biallelic deletion of E-cadherin. This is an event that is incredibly rare in invasive ductal cancer and suggests that this cell line is more than likely came from an ILC and is probably misannotated. It is genetically likely to be ILC. And I think this is the power of the optical mapping. It gives you this incredible resolution and mapping over long ranges of these complex rearrangements, both the chromothripsis, but then these singular genetic deletions. This is another one. This is also SKBR3, which is labeled at ATCC as an IDC cell line, but it phenocopies ILC and looks ILC-like. When you look, this again has one allele is lost, and on the second allele, similar to the cell line I showed you previously, it doesn't delete CDH1, it actually deletes CDH3, the gene that's next to it. This is a similar gene to CDH1, and that deletion actually comes halfway through CDH1, fuses it to this gene down here, and this becomes non-functional. So basically, similar, it has biallelic disruption of CDH1, similar to the previous cell line. So to summarize, these DNA structural aberrations are common in breast cancer, and they have prognostic and therapeutic value. Optical mapping provides genome-wide information on all DNA structural aberrations in a single assay. The copy number aberration data is concordant between SNP arrays and optical mapping, telling you that it's measuring essentially the same thing. Chromothripsis, chromoplexy, and contagious are common in cell lines and tumors. I didn't show you all the data we've seen with the optical mapping, but basically you can resolve a lot of this using this large-scale mapping. And optical mapping detects complex loss of heterozygosity and structural aberrations at the CDH1 locus in both ILC and these ILC-like cell lines, suggesting that some of these ILC-like cell lines more than likely probably are true ILC cells. I would just like to acknowledge I run the laboratory with my wife. This is Steffi Oostenrijk. She's also a professor in pharmacology. We run the lab together. These are the lab personnel. We're lucky to have a number of great students. We have an incredible collaboration with our physicians, and we thank obviously the patients who've donated tissue to much of our research. And finally, thank Bionano for use of the optical mapping platform. Thank you.
Hello, everyone. My name is Justin Balco. I'm an associate professor of medicine and pathology, microbiology, and immunology at Vanderbilt University Medical Center and Vanderbilt Ingram Cancer Center. Thank you very much to Bionano for having me give this presentation on some of our work together. The title of my presentation today is Optical Genome Mapping of Large Structural Variations in Non-Inflammatory and Inflammatory Breast Cancer. So we, for a while as a lab, have been very focused in breast cancer, and particularly immunology of breast cancer. And for a while now, also interested in this one very rare subtype of breast cancer called inflammatory breast cancer. So inflammatory breast cancer is defined as a clinical subtype, and it has a particularly poor outcome. So it has a very early time to metastasis in patients. It seems to have a disproportionately poor response to chemotherapy, followed by complete mastectomy, or it requires complete mastectomy in many cases. And it's disproportionately hormone receptor negative. It has high rates in African-American patients as well. Although importantly, inflammatory breast cancer can encompass all types of breast cancer, including hormone receptor positive, HER2 positive, and triple negative breast cancers. So the clinical signs that define inflammatory breast cancer are what pathologically we would call T4D, which means metastases to the local region, the skin. Also a feature called peau d'orange, which is an orange skin-like inflammatory effect on the skin near the breast, near the site of the primary tumor, as well as mastitis-like symptoms, which often result in a very difficult diagnosis, or in some cases a misdiagnosis. Pathologically, we define inflammatory breast cancer as having these dermal lymphatic invasions of tightly clustered tumor emboli. And what you'll see here on this H&E stain is that in the dermis of the skin, pathologists will identify these clusters of tumor cells that are migrating out from the primary tumor and invade into the lymphatic system, causing these swelling and the peau d'orange and clinical symptoms that I described previously. So for a long time, researchers have been interested in the genomics of inflammatory breast cancer. There's been several studies on the genomics and transcriptomics that have been performed and reported in the literature. However, many of these studies, due to the rare nature of inflammatory breast cancer, have been heavily confounded by subtype imbalance, small numbers of patients, poor or non-existent false discovery rates for identifying features, and some modest enrichments for pathways, which are already really known to be associated with technical noise or triple negative breast cancer, HER2 positive subtypes. If you recall, inflammatory breast cancer, while it includes all of these subtypes, seems to be biased towards TNBC and HER2 subtypes. There's been a number of different techniques used: RNA sequencing, targeted next generation sequencing, exome sequencing, and most recently whole genome sequencing. The unique and very distinct clinical and pathological features of inflammatory breast cancer suggest that there's an underlying pathognomonic alteration. What I mean by that is some kind of genetic alteration that is really synonymous with this feature of inflammatory breast cancer that really defines this biological subtype. And so we hypothesized that since all of these things have been done really to no avail or no defining feature, one poorly captured area by these prior techniques that remains to be explored would be large structural genomic variations, which are really quite difficult to detect with any of those other technologies. And so in order to assess this, we utilized the Saphyr platform to test the hypothesis. This is just a data summary, initial set of samples. I know you guys are familiar now with the technology from prior presentations. Our initial set that we ran on the Saphyr instrumentation or pipeline included 2 inflammatory breast cancer cell lines, some 149 and 190T, and an actual clinical IBC sample, 2 comparator triple-negative breast cancer primary breast cancers, and then an inflammatory breast cancer patient-derived xenograft model. And so if you look at these summary statistics, you'll see that they're quite similar for the detection, for the number of data molecules collected, and the different types of variants that we identified within each one of the samples. So no huge biases within this initial data set. These are just some of the Circos plots showing the structural variations that we identified. And so what I want to really get to the point on here that's of big interest to us is that we found, of course, many different types of large structural variations in these samples, but most specifically, one of the things that interested us was something that hadn't really been defined in any breast cancer prior, and those were deletions or large structural variations in the ODS2 gene. And those are identified here. And you'll notice that they occurred in one of 2 different IBC cell lines. They occurred in the clinical IBC sample and in the patient-derived xenograft from an inflammatory breast cancer patient as well. We also did identify one ODS2 insertion and duplication in one of our controlled non-inflammatory triple-negative breast cancers. Now, if we take a little bit closer look at these ODS2 large structural variations we identified, you'll notice that we've outlined where the introns and exons of ODS2 are here in chromosome 7. What was really interesting was that the non-inflammatory breast cancer in which we identified an ODS2 variation in, shown here, was actually in one of very large intergenic regions. So at least as far as we know, this wouldn't be predicted to alter any kind of gene transcription or function. However, for all of the other IBC samples where we identified ODS2 lesions, each of these included at least one exon. And in many cases, at least in the clinical samples, very large deletions of many exons towards the C terminus of the gene, or the 3' end of the gene. So just to show you some of the data supporting this, you can see that there's a large number of molecules here that really support the deletion of the the C terminus of the ODS2 gene. These are reads or molecules that were identified from the PDX model that we incorporated into the analysis. And you can see that the copy number here really does show that there seems to be something going on. So both the copy number analysis as well as the molecule support seems to show that there's a deletion here in the C terminus. So what is ODS2? Really, ODS2 has not been studied in the context of cancer at all. This is some pictures from what's called ODS2 syndrome. So it's actually been described as being a familial or a genetic, when you say disease, a syndrome rather, that results in low birth weight, feeding difficulties, intellectual disabilities, and microcephaly and mild dysmorphic features. So it's called ODS2 because it was originally thought to be associated with an autism-like syndrome. Other work that's been done in this field has suggested that when looking at exonic and whole gene deletions in large data sets of patients, both control and patients with ODS2-like syndrome, that the greater severity of the disease or the syndrome seems to occur just like what we identified with deletions that are lying in the C terminus of the gene. And there's an inference from studies that there seems to be a haploinsufficiency without a dominant negative function. What I mean by that is the single copy loss seems to suggest, or where data analysis seems to suggest, that a single copy loss in the germline can cause the syndrome. So this has also been studied in mice, and this seems to, whenever ODS2 is deleted from the germline in mice, as you can see here, this results in many of the same, at least dysmorphic features that we observe in patients. And this paper here that appeared in Nature a few years ago really helps define OS2 as being an epigenetic modulator as part of the polycomb repressor complex that can alter this from a repressive to an activating phenotype, the presence of OS2. So OS2 is mainly, as I needed to mention there, is really thought and really only studied to be expressed within brain tissue. So the first thing we wanted to do is validate this. We were able to develop, at least specific to the human IBC sample that we analyzed by Saphyr technology, we were able to develop a FISH probe that would cover that region. And as you can see here, looking at the copy ratio, we actually do see what appears to be a single copy loss in about 50% of the cells within the tissue sample by FISH. In a second analysis, we also performed Saphyr pipeline analysis, rare variant pipeline analysis on a series of non-inflammatory cases. We really wanted to make sure that this wasn't a technical artifact or it wasn't at least something that was very common in breast cancer that hadn't been picked up. So all these are non-inflammatory breast cancer cases. We did 8 different samples. Just really quickly going through the findings here, we accidentally included, I hate to say that, but we accidentally included 2 different regions of the same tumor from the same primary resection that had different sequence ID numbers. So there's a little bit unfortunate, but it gave us a real opportunity to look at reproducibility of the assay going through DNA isolation, different regions of the same tumor, and then also the data analysis. So you can see the high degree, striking degree of similarity between those 2 samples, demonstrating some level of reproducibility. We can also see things we expect. So to some degree, in HER2 amplified tumors, we can see either low or high degrees of amplification, including chomothripsis in the HER2 positive sample over here on the bottom right. And in this secondary analysis, we did identify one more deletion in a triple negative non-inflammatory sample. And this was a very small intronic deletion. So again, not disrupting presumably gene function. And so our conclusions and future directions are that the genomic underpinnings of inflammatory breast cancer still remain to be discovered if they actually do exist. And hopefully elucidation will aid us in not only better treating this aggressive disease, but really understanding the biology behind it. We did identify OS2 deletions that involve the C-terminus domain that seem to track with inflammatory breast cancer status, but we need to be able to validate this in larger sample sizes. And we also would like to determine whether deletion of this specifically in tumor-bearing models, such as the new IRIS-CRE model, would generate any kind of inflammatory breast cancer features. And this would be an incredibly useful genetically engineered mouse model if this is the case. So we're currently in the process of creating floxed mice that can be manipulated to lose OS2 specifically in breast tumors. So with that, I'd like to acknowledge some of our funding, many of the things that put this project together, including the Bionano Genomics team, our pathologist here, Paula Gonzalez-Erickson, and then a donor of ours, Terry Arnold, who is an inflammatory breast cancer survivor herself, and has really done amazing work in the last 15 to 20 years developing the IBC Network Foundation. We also received funding from Genentech Roche and NIH. And then with that, I would like to close, and feel free to email me any kind of questions. Thank you.
Hello, my name is Eric Letouzet. I am a computational biologist at the Cordelier Research Center in Paris. And today I'm going to show you how, by combining whole genome sequencing and Bionano genome imaging, we could unravel complex structural rearrangements induced by replication stress in liver cancer. In the lab, we are interested in liver cancers, and in particular, hepatocellular carcinomas, which is globally the 3rd cause of cancer death. This cancer has a heterogeneous incidence in the world related to the diversity of risk factors. Most cases occur in sub-Saharan Africa and Southeast Asia related to HPV infection and aflatoxin B1 exposure. There is also a high incidence in Japan and Egypt due to HCV infection. In France, alcohol is the main cause of the disease. And in the US, metabolic diseases are the principal risk factor. These risk factors operate by damaging the liver, and it is known that more than 80% of HCC develop in a cirrhotic liver by the transformation of dysplastic nodules. However, a little less than 20% of the cases develop on a non-cirrhotic liver. In this case, one of the main transformation mechanisms is the activation of oncogenes by viral insertions or structural rearrangements. In particular, we recently described a new molecular entity of HCC driven by viral insertions and rearrangements activating cyclins A2 or E1. We found that tumors with activated cyclin A2 or E1 define a homogeneous molecular group of HCC, as you can see on these gene expression-based classifications. They also have specific clinical features, in particular a poor overall survival. There are also large tumors, usually bigger than 10 centimeters, occurring in relatively young patients and in absence of fibrosis. At the molecular level, these tumors, called CCNHCC, display a high frequency of RB1 and PTEN inactivation, as well as a striking activation of ATR pathway implicated in replication stress response. Indeed, cyclins E and A are implicated in the entry and progression into S phase. And it is well known that when you activate cyclin A2 or E1, you will induce premature S phase entry and replication stress. Then we wondered what kind of genomic instability could be induced by this replication stress phenotype in this tumor subgroup. So we sequenced the whole genome of 50 tumors comprising CCNHCC and tumors of other molecular groups. We found no difference in terms of point mutations and indels, but we found that CCNHCC display a very high number of structural rearrangements. In addition, when looking at the nature of these rearrangements, we found that replication stress in these tumors induces a specific signature, the RS1 signature, which is characterized by focal tandem duplications coming together with a lot of translocation breakpoints. This phenotype is easily recognizable on a circus plot. Here you can see the abundance of tandem duplications in green and interchromosomal translocation breakpoints in gray. And it also leads to a particular copy number profile with hundreds of focal gains distributed throughout the genome. To get a deeper understanding of this phenotype, now we can zoom on one particular chromosome. Here we are. And now we can see in more details those very focal gains that are very clear on the log ratio profile. And on top of this log ratio profile, we overlaid the abnormal junctions identified by short-read whole genome sequencing. In some cases, the focal gains are induced by classical tandem duplications, leading to the duplication of one segment adjacent to itself. But in other cases, like here, a focal gain is actually surrounded by interchromosomal translocation breakpoints, here going to chromosome 2. And if you go to chromosome 2, you will see another very focal gain with interchromosomal breakpoints going back to chromosome 14. This kind of event, typical of this tumor subgroup, is called templated insertion cycles and is characterized by many focal gains being interlinked with interchromosomal translocation breakpoints. Now, it's very difficult with short-read whole genome sequencing to figure out precisely what this complex event looks like. All we can do is make hypotheses And we found two hypotheses that could explain this kind of observation. One in which the E segment would be inserted between two copies of the B segment inside chromosome 14, and another equally likely possibility in which the B segment would be inserted in the middle of two E segments on chromosome 2. And because there's no way with whole genome sequencing to demonstrate the existence of these events and to discriminate these two possibilities, we decided that we would analyze a few cases with Bionano. So we analyzed 4 HCC samples and matched normal liver with Bionano optical mapping technology, starting from 30 milligrams of frozen tissue. We successfully obtained high molecular weight DNA, more than 150 kb long, for all but one sample for which we did not provide enough starting material. We obtained a genomic coverage around 300x with an average molecule size of 250 kb. So this was a very important step, and we were happy to obtain, to be able to extract really long DNA molecules, because with our classical DNA extraction kits, we usually get DNA fragments of 10 kilobases. From this data, we analyzed structural variants using Bionano software. And because we are interested in the somatic structural variants that are specific of tumor cells, we made a detection in both the tumor and matched normal sample. And we compared the two SV call sets to be able to extract only the somatic structural variants that occurred in tumor cells. We then represented the global profiles of structural rearrangements with circus plots, as you can see here for the 4 tumors. The 2 tumors on the left are CCN HCC tumors driven by HPV insertion in cyclin E1 for the first and cyclin A2 for the second. And the 2 tumors on the right are tumors without activation of these cyclins. What we can see here is that we are able, with Bionano technology, to retrieve our RS1 signature of replication stress with plenty of tandem duplications and interchromosomal translocations. Interestingly, in the second sample, we knew that we had some tandem duplications that were very focal in the range of 10-20 kilobases. And for these events, usually you don't have enough DLE markers to be able to map with precision the segment on the genome. Yet, Bionano software did identify those events as insertions, despite their very small size. So the RS1 signature is detected exclusively in the 2 CCN HCC samples. Next, we compared the structural variants called with whole genome sequencing and Bionano for 3 tumors that were analyzed with both technologies. Whole genome sequencing was performed as quite a high depth because we had a 90x coverage in the tumor and 30x in the normal samples. On this slide, you can see the number of structural variants per category identified by whole genome sequencing on top and Bionano at the bottom. The first tumor on the left is relatively quiet in terms of structural rearrangements, whereas the 2 others correspond to cyclin HCC. What you can see overall is that we have a very good match between the 2 technologies, with the majority of structural variants identified by both. However, for each tumor, we have some events identified only with Bionano and whole genome sequencing. Globally, we have more events identified with Bionano, on average 1.5 times more events than with whole genome sequencing. And we also have a larger diversity of events, including some intrachromosomal rearrangements. And we also noticed that some of the tandem duplications are classified as insertions. These correspond to the very small events. So we had a look at the events that were found only with whole genome sequencing, and they generally consist in very small events, smaller than 10 kilobases. And then we had a look at the events that were only detected with Bionano, which you can see on the next slide. Here, we have the coverage log ratio profile of one chromosome that is represented. 1 point corresponds to 1 kilobase. And on top of that, we overlaid the structural variants identified by whole genome sequencing on the top and Bionano at the bottom. We can see that many events are only detected with Bionano. These include centromeric events. It is known that these regions contain repetitive elements and are difficult to analyze with sequencing technologies. Others correspond to subclonal events that we detected due to the very high coverage we had in Bionano data. And finally, a lot of events correspond to highly complex rearrangements that were missed with whole genome sequencing approaches. Here, the red arrows, for example, point at a series of intrachromosomal rearrangements that bring together distal junctions and lead to a very complex rearrangement. And most of these junctions were missed in short-read sequencing. So now let's go back to our typical events induced by replication stress, which we wanted to characterize with optical mapping. Like I said, for this kind of interchromosomal templated insertion cycle, we cannot really detect the precise event with whole genome sequencing. All we can do is make hypotheses. In this case, there are two hypotheses, either segment B within 2 copies of segment A or segment A within 2 copies of segment B. With Bionano, we could obtain long molecules spanning the full events. On this graph, you can see the molecules at the bottom and the map reconstructed from these molecules in gray. And if we go through this map, we can see that it starts with the first segment of chromosome 3. Then we have the B segment that is duplicated at 2 positions. And in the middle of these 2, you will see the A segment from chromosome 1, and then the end of chromosome 3. So here we can confirm for the first time the existence of these templated insertion cycle events, and we can identify unambiguously the host chromosome, in this case, chromosome 3. We also identify other kinds of events which are strongly related but involving only intrachromosomal templated insertion cycles. In this case, we have 2 gained segments linked by inversion breakpoints. And with Bionano, we identified long molecules validating the event in which segment A is inserted into 2 copies of segment B at the end of chromosome 10. Finally, we could reconstruct very complex events involving several segments, like this one involving 3 segments on chromosome 3 and 1 segment on chromosome 6. With Bionano, we could unambiguously identify the complex event that involves the insertion of segments C, A, and D in the middle of 2 copies of segment B located on chromosome 3. So overall, we have a diversity of structural rearrangements related to replication stress. About half of them are classical tandem duplications, and another 40% are templated insertion cycles that can involve intrachromosomal or interchromosomal events. All these events that represent 82% of alterations in this subgroup can be explained by a break-induced replication mechanism in which the replication machinery is stalled at some point, will jump to another location, replicate a stretch of DNA, and finally come back to the original site of replication. In conclusion, we identified a subgroup representing 7% of HCC, driven by cyclin A2 or E1 activation, that will induce premature S-phase entry, replication stress, fork collapse, and this particular signature induced by break-induced replication repair. This gives rise to large tumors, very aggressive, but this replication stress phenotype suggests these tumors could be targeted by PARP inhibitors or ATR pathway inhibitors. In terms of what Bionano helped us to do here, it allowed us to reveal more structural rearrangements than whole genome sequencing, complex events induced by replication stress, and it may be used to identify replication stress in clinical samples. I will finish by thanking my sponsors, my colleagues in Jessica Zickman-Ross's lab, in particular Quentin Bayard, who was the main PhD student involved in this project, and our collaborators abroad. Thank you for your attention.
I'm not sure if this is an intermission or what's going on here. I'm gonna give it a couple minutes and yep, there it goes.
Hello everyone, I am David Joancien, manager of the Genomic facility in the Translational Research Department. Today I will present you some results I obtained during my PhD concerning genomic structural variant which lead to specific DNA scaffold and fusion RNAs in our aggressive uveal melanoma models. So uveal melanoma is a rare disease, less than 600 cases per year in France, and representing also less than 5% of all melanomas. This tumor occurring in the eye is mostly located in the choroid layer, followed by the ciliary body and the iris. The primary tumor is a local disease and under the control of surgery and proton therapy. The main point concerns the metastasis occurring in around 50% of patients. Metastases are mostly located in the liver, followed by the lung, skin, etc. Main mutations are in the GNAQ11 genes. Those mutations are activating mutation, activating the GPCR pathways, and in this pathway, PLC beta 4 and cis-LRT2 can be also mutated. A second event is present in most of uveal melanoma cases. In 80%, in fact, BAP1 or SF3B1 or EF1AX are mutated. This is a mutually exclusive mutation, in fact. And concerning genomic alteration, Nathalie Cassou, our surgeon, published in 2014 an updated classification of based on the genomic alteration and the structural abnormalities concerning chromosome 3, chromosome 6, and gain of the IQ. And depending on the loss of the chromosome 3, the gain of the 6p and IQ mostly, the outcome is different, and the high risk is observed in the case where a monosomy 3 and a gain of the IQ is observed. Same result has been published by the TCGA effort. As you can see, BSC events and specific copy number alteration also propose a four-class and four-group classification. Sorry. In order to improve our knowledge of uveal melanoma, and because of BAP1 mutation. For example, we've decided to dig into the epigenomic field, and we've decided to study the DNA organization in order to link this analysis to the gene expression. To achieve this goal, we've decided on specific models to achieve a specific whole genome sequencing analysis coupled to a whole genome bisulfite sequencing to study the methylation, the DNA methylation. We also had the analysis of histone modification mark, the gene expression, and the DNA topology thanks to the in situ IC methods. Since 2010, since more than a decade, we study different ways to have models for uveal melanoma, and thanks to Fariba Nemati, who set up a lot of models, grafting nor primary tumor or metastatic tumors. We've characterized different kind of models which recapitulate the different features of And together with Fariba, we've selected two different models, aggressive model, in fact, MP41 and MP46. Those models have specific features concerning the status of the chromosome 3, the 6, etc. But still, they share a gain of the AQ. They don't have the same chromosome 3 status. MP46 has a loss of the BAP1 protein expression. The two models have a mutation nor in GNAQ or in GNA11. And thanks to the lab of Professor Simon Saul in Curie-Orsay, and thanks to Géraldine Liu, I was able to compare those models to normal choroidal melanocytes. And this was a very nice opportunity for our characterization. The first step of my PhD was to isolate the cells from fresh tissues, and to do that with the help of Ronan Crépin, with designed and validated nanobodies in order to sort the tumor cells. Specifically, with specific nanobodies, we were able, in fact, to sort a lot of cells. Thanks to that, we've done the RNA-seq analysis, identifying more than 4,000 regulated genes. And this list of genes was compared to the specificities of the genome, its organization, the compartment, exon, and so on. And recently, we've added to our analysis optical mapping and MFISH to confirm our results. So because the in situ IC experiment takes a lot of time, requires a lot of cells, and this is expensive experiment, we've decided to apply the specific pipeline, a bioinformatic pipeline, dedicated to the identification of structural variant and translocation. Nicolas Servant, our bioinformatician in Curie, applied this on our models and identified more than 1,000 translocations. Thanks to the whole genome sequencing dataset, this list was reduced to 50 translocations in MP41 and MP46. Those translocations were located in several places of the genome, but some were interesting for causing fusion RNAs and fusion proteins. So I tried to validate all the translocation using targeted PCR approaches, but unfortunately, I wasn't able to confirm any of the 50 translocations. And finally, thanks to the optical mapping, I wasn't able again to find those 50 translocations. So this analysis wasn't successful at the end. During the validation of the translocation, in fact, we did RNA-seq specific analysis, thanks to Pierre de Lagrange from Genosplice, we applied 5 different algorithms for the fusion prediction. We keep at that time the fusion that were found in at least 2 algorithms and present in at least in 2 replicates per model. So among the 318 fusion RNAs, 14 fusion RNAs were kept and validated as present in our aggressive uveal melanoma models. And they were also associated with structural variants or due to specific DNA contact. So this is a summary of this analysis, but I will present you some example of those fusion RNAs. So here is an example of MAP-kinase 5 and ACAT10 fusion RNAs. This RNA was identified in MP41. MAP-kinase 5 is located on 12q24, just after ACAT10. And as you can see here, after the 7th exon of MAP-kinase 5, the fusion RNA is fused to exon 14, and this gray box corresponds to the sequence. I've done the PCR validation and the sequencing that prove that this fusion RNA is present in our sample, and this is the sequence of the fusion protein that could be produced. And going back to the DNA topology, we can see that in normal melanocyte there is a subtad there, and surprisingly in MP41 this subtad is modified. And thanks to the optical mapping, we saw that the 2 alleles were present in MP41. The first one, this blue track, is normal and corresponds to the reference here in green. As you can see here, this pink box embracing ACAT10 to MAP-kinase 5 is in fact duplicated on the second allele there, duplicated and inserted just after the 7th exon of MAP-kinase 5. And this structural variant led to a specific fusion gene from MAP-kinase 5 and ACAT10 and produce after probably a fusion RNA. So thanks to the optical mapping, we can see the coverage on each single allele. More than 60x can be seen on that picture. And in fact, this fusion RNA was previously detected by people working on autism, and Pierre de Lagrange also identified this fusion RNA, into TCGA samples. So to summarize this structural variant, this insertion duplication in ACAT10 MAP-kinase 5 modified the chromosome conformation, led to a specific fusion gene, and led to a fusion RNA in MP41. Here is an example of another fusion RNA that was identified between cabin 1 and MRPS 21. Cabin 1, in fact, is present on chromosome 22, and MRPS 21 is present on chromosome 1. So first, I've confirmed this junction from exon 10 and exon 2 of the 2 genes. This was in agreement to the RNA-seq, and in fact, this fusion RNA could lead to a specific fusion protein again. Going back to the optical mapping, we can see that there is a contig here in light blue sharing a piece of the DNA from chromosome 1 and the other part here from chromosome 22. And more precisely, the breakpoint is in the middle of MRPS 21 from chromosome 1 and in the middle of cabin 1 from chromosome 22. And thanks to the MFISH analysis, so here this is the multicolor painting, you can see an equilibrated translocation between chromosome 1 here and chromosome 22 in red And as you can see, it's equilibrated. And furthermore, going back to the contact map, we can see specific contact in MP41 close to the breakpoint and concerning the localization of these two genes. And this fusion RNA has been identified also in 3 cases of uveal melanoma from the TCGA. And I would like to present you a first example of structural variant that is a bit amazing for me. So we identified two fusion RNAs. The first one from GP84 and NCOS7. So going to this contact map, so here this is the chromosome 8, where GP84 is located. And this gene with NCOS7 on chromosome 6 give a fusion RNA, and here the letter underlined are corresponding to the junction of the fusion RNA. And there is in the same place, there is 2 other genes, POMK, also present on chromosome 8 here, with RSPO3 on chromosome 6. Together, they produce a fusion RNA. The sequence here corresponds to the junction. So this is validated after sequencing. And this fusion RNA can also lead to a fusion protein. So thanks to the optical mapping, we can see that there is a translocation between chromosome 6 and 8. The breakpoint is occurring from the chromosome 6 in the middle of TEMIS and close to SFRP1 on chromosome 8. So as you can see here, this translocation is not so far from the 4 genes we spoke before. But taking a wire to try to have a model of what happens there, probably this new DNA fragment coming from the fusion of chromosome 6 and 8, so here is the breakpoint, probably the transcription starts from this era, from GAPT4, and the enzyme jump to NCOS7 to produce a fusion transcript. And possibly the same occurs on POMK. The enzyme starts on POMK and finish on RSPO3. This could be an explanation of the fusion RNA. So this is not like read-through RNAs or transplicing, but because of this high number of contacts there, we think that this could lead to specific fusion RNAs. Nevertheless, going back to the contact map between chromosome 6 and chromosome 8, we went back to the MFISH and we saw that there is a complexity of the genome, like a chromotripsis, because when we look on chromosome 6 or on chromosome 8, and when we label specifically with for chromosome 6 or 8, in fact, here in the middle, we don't see that clearly here, but there is a thin layer coming from the chromosome 8. On this chromosome 8, there is this is corresponding to an isoderivative chromosome 8, with in the middle a piece of chromosome 6. And again, concerning the chromosome 1, we can see 2 dicentric chromosome 1 with a piece of chromosome 11 there, and next a piece of chromosome 6, as you can see here, and a piece of chromosome 8. So this gives probably a specific genome instability, and this could explain the centric ring formation and the ring chromosome that we observed in our model. So probably we think that this translocation may produce different derivative chromosomes 6 and 8. with additional modification and give specific abnormalities in the ring chromosome. So to conclude, in fact, we've identified specific fusion RNAs with a non-function yet. Those fusion RNAs are coming from complex genomic abnormalities, which were observed thanks to the MFISH and the optical mapping. Concerning the next step, I will confirm the presence of this translocation in the primary tumor and in the early passages of MP41 using specific probes that targets the 4 genes of interest. And also, in order to dig into the functions of the regulated genes and on the fusion RNAs, we will do specific data mining and text mining, thanks to the tool that Sébastien Tourlet designed previously. So thanks a lot for your attention. I would like also to take the opportunity to thank all my colleagues, especially my boss, Sergio Roman, Fariba Nemati, Josh and Elnaz, who make the integrative analysis, as Pierre Delagrange is doing right now on ChIP-seq and so on. I would like also to thank my colleagues from the genomic team, Sébastien Tourlet and Radia, who did a lot for the MFISH. And I would like also to thank the different members of Bionano who first helped me to perform the first analysis on Bionano in Clermont-Ferrand on the Gentian platform. So thanks to Michaela and Charles Poncet. But also thanks to Bionano, I was able to combine the whole genome sequencing and the optical mapping with the help of Yuval Porat from Genox. So this collaboration was very helpful in order to improve the comprehensive analysis we were doing on uveal melanoma. Thank you very much.
Hello, everyone. Welcome to today's symposium on solid tumors. My name is Ravi Kohli. I'm a pathologist at Medical College of Georgia at Augusta University, and I'm going to talk about our experience of using optical genome mapping on Saphyr for assessment of solid tumors, especially adult brain tumors, because that's our tumor of interest. These are my relevant disclosures. I'm employed at Medical College of Georgia at Augusta University, and these are some of my research support from industry. And most importantly, the opinions expressed during this presentation are my personal opinions and did not reflect or represent the opinions of any of my collaborators or employers. And the device or the instrument I'm going to talk on today, the Saphyr, is intended for molecular biology application, and this is not intended for diagnosis, prevention, or treatment of disease. This is my outline. In the next 20 minutes, I want to take you all on the journey of our experience of working with the Saphyr optical genome mapping on adult brain tumors. A little bit of background about who we are. So my lab, I was recruited here 8 years ago to develop the molecular pathology program, and the lab which we have designed and developed is a CLIA lab, which is created by the state of Georgia for high complexity testing, and the faculty in my lab is mostly pathologists and doctoral scientists. In the last 8 years, we have put together a pretty solid molecular program, which encompasses most, if not all, the new technologies and platform we need for the new generation of molecular testing in a clinical setting. So if you look at our journey at cytogenetics movement to molecular cytogenetics, it was in 1988 we termed the chromosome, the term chromosome was coined. And as we moved away from this particular incident, then you can see the fish was added in 1986. The arrays were started, and we received the first FDA clearance for chromosomal microarray in 2014. And one of our plans going forward from movement of molecular cytogenetics to molecular cytogenomics is to work on newer technologies and platforms like optical genome mapping and see where and how we can incorporate that platform and technology in clinical care, especially both the constitutional as well as tumor types. Yesterday, I think you heard about the heme malignancies, and today we're going to focus mostly on how we can use optical genome mapping on solid tumors. So as we all know, karyotype still is the gold standard for chromosome analysis, but it has its own challenges. It has a very low diagnostic yield. The other major challenge we have is the very long turnaround time for solid tumors for the culture growth. It definitely misses the small changes, most commonly less than 5 megabase. We really need very special trained people in cytogenetics. I think that's becoming a major commodity now. And it is because it's a subjective method, it's prone to human error. So with this background, I want to move to our journey of using optical genome mapping in solid tumors. Mostly, I'm going to focus on adult brain tumors because that's one of the most interesting tumors we have identified as one of the phenotypes we want to work on. So I'm a pathologist by training, so I have to bring in some H&E slides, even though this is a cytogenomics symposium. So adult brain tumors traditionally, for all these years and decades, have been classified into grades, grade 1 to 4. And this is how normal brain tissue would look like. And low-grade tumor and high-grade tumor, as you can see, mostly depending on the site as well as the cell type of origin. You can have a pilocytic astrocytoma, ganglioma, a pleomorphic xanthosarcoma, which probably will fit in your low-grade tumors. And on the high grades, you have the diffuse midline gliomas, the medulloblastomas, and primitive neuroendocrine tumors, which would come under the high-grade category. So this, again, was a very gold standard way of not only diagnosing adult brain tumors, but also for prognosis and therapy selection. Low grades, better prognosis; high grades, worse prognosis. But there was a paradigm shift in this understanding of these low-grade gliomas, especially based on the TCGA analysis in 2015. What we were able to identify that the diffuse low-grade glioma, especially the grade 2 and 3, we really need to look at the molecular analysis, especially the IDH1 mutation as well as 1p19q deletion. And based on these two main molecular abnormalities, this clinical presentation is very different. There is literally split survival based on these two tumors, irrespective of histologic subtype. And there are definitely additional molecular alterations do play a role on top of 1p19q co-deletion along with IDH1 mutation in these low-grade diffuse glioma, especially grade 2 and 3 histologically. So the distinction, the clinical outcome was so significant that if you compare the molecular histological classification versus molecular classification, you can clearly see that a low-grade glioma with the IDH mutation and 1p19q deletion has a pretty split survival as compared to a glioblastoma with a wild-type IDH. So what dictates basically is not only a histologic subtype, but the presence of molecular dynamics in these tumors. And that has led us in the last 5 to 6 years to identify, on top of the histological subtypes, lot of molecular information we would need to classify these tumors. So in summary, adult brain tumors are relatively easy to diagnose on histology as well as on biopsies. Not only diagnose, but also the grade. But based on the TCGA data, we have identified that especially the WHO grade 2 and 3 gliomas have a split survival, and this has led us to go into the molecular analysis of adult brain tumors for diagnosis as well as prognostications. So there are multiple papers which have been written in the past as well as been adapted. So we went through a whole bunch of published articles and their genomics on top of the TCGA data set, and we put together our own algorithm of looking at these tumors in the lab. This is what we clinically do on a daily basis using a SNP array to make that distinction of diagnosis as well as prognostication based on IDH1 mutation as well as 1p19q codeletion, along with the whole variety of other genomic aberrations. So with that history in the background, I want to take you guys to our plan to look at these adult brain tumors which we have banked and see how optical genome mapping, number one, help us concordance with existing molecular data we have. And on top of that, what additional information would OGM would add to it, especially any clinically relevant information. So we are taking out 30 archived histologically diagnosed adult brain tumors from our tumor bank, and we want to make sure that all these tumors have known molecular testing done, especially the OncoScan, which is a SNP array. On top of this, most if not all the tumors will have done P53 IHC, IDHR1 IHC, 1p19q deletion by FISH, EGFR amplification. So these are all fresh frozen tumors biopsies from the glioblastomas. OncoScan has performed or will be performed on the exact same DNA on which will be the optical mapping. All the samples tested on the OGM were blinded to OncoScan data, and the annotation was done based on the clinical SOP. Apart from other additional information that we had identified based on the evidence-based pathology as well as meta-analysis and the TCGA data, we had our initial investigation focused on clinically relevant genomic aberrations which are very well established in the adult brain tumors. And this is just a few examples of them which we want to make sure that we do identify the status of these aberrations in these brain tumors, which we have done. The solid tumor study sample prep is directly from the Bionano kit protocol. We used around 5 to 20 milligram of fresh frozen tissue for DNA isolation, and we used the Bionano Prep SP tissue DNA isolation kit in the lab for this particular study. The Saphyr workflow, if you are not aware of it, and the key thing is isolation and characterization of high molecular weight DNA, labeling of that entire genome, transferring the labeled DNA to the cartridge, and then assembly and analysis of the genome. I want to take you through some of the really cool cases which we have investigated. This is probably the very early insight and an adult brain tumor, especially in compared to OncoScan. This is the case number one. This is an adult white male with a central glioblastoma. And here you can see that the slide shows a CNV profile by OGM compared to the SNP microarray. You can see the SNP on the bottom OncoScan and the OGM on the top. The top panel is OGM data and the bottom is microarray. Both the profiles shows the concordant aneuploidies and the CNVs, especially in chromosome 2, 6, 7, 10, 14. 19, and 22. I want to dive deeper in that data. And one of the major advantage of using an optical genome mapping technology is identification of an amazing discovery information, which is impossible on a karyotype or array, which is the fusion. And this is probably the poster child case for looking at adult brain tumors on optical genome mapping, and we were able to identify a variety of complex translocation along with all the clinically critical genes, and in addition to that, a lot more clinically important genomes. So the circus plot basically shows you the copy number, the cytoband, and interstitial SVs, the translocation, and an aneuploidy. And these are all the abnormalities which are absent in the Bionano control sample structural variant database, and this is how the pipeline works. And the first table I showed you earlier, the clinically important genes are highlighted in black. We want to make sure that we do definitely see some abnormality, yes or no, in those genes. And then I'll show you the genes that are unique to the Bionano database. So this is that earlier tables of what I showed you, which is expanded. We are going not only by chromosome, but also looking at the gene level to see if there is any CNVs, SVs, or complex abnormalities, and then comparing with our standard of care OncoScan data. And this, and the red boxes are the ones which we have identified only in optical mapping, and the black boxes basically shows you the concordance with concordance of optical genome mapping with OncoScan on case-by-case basis at a chromosomal and gene level. Continuing on the same case, here what we're trying to show is a mosaic homozygous deletion of CDKN2A. The circus plot on the top right shows chromosomes 7 and 9 with unbalanced translocation, which was indicated in the OGM, and the high resolution of the nested deletion, which you can see on the copy number profile with approximately 1.3 copy number state for that particular chromosome, that particular gene. And this is a result of unbalanced 7 to 9 translocation and an additional nested deletion. The unbalanced translocation is beyond the detection of SNP microarray because that's a major limitation. And as I said, not only we are able to get 100% concordance with the SNP microarrays, but on top of that, we are identifying a pretty significant clinically, potentially clinically relevant information, which was not there, what was not there identified in earlier, even the TCGA database, because most, if not all, the TCGA database is done on OncoScan. So looking at these tumors, it might be very interesting to go forward and compare that with the TCGA database. And continuing on the same sample or the same tumor, this is an MDM4 amplification, which is uniquely called by optical genome mapping. The chromosome 1q33, the MDM4 is amplified around 14 copies. And the circus plot here shows 14 copies of MDM1 amplification. And on top of that, there's a fusion with chromosome 2, 12, 16, and 17. I think this is another very interesting information which we have identified in this particular tumor. And the panel on the bottom shows multiple abnormal fusion of chromosome 1, which are involving in and around the MDM4 gene. We're still further investigating the clinical relevance and importance and even existence of these translocation, not only brain tumors, but also other solid tumors. So this is a case number 2, which shows this shows a slide comparing the CNV profile by OGM to SNP microarray. The top panel is optical genome mapping, and the bottom is microarray. And both the profiles show concordant aneuploidies in chromosome 1, 4, 5, 7, 8, 10, 13, 14, 19, 20, 21, and 22. So even at the whole genome level, you can clearly see a distinct concordance with overall whole genome abnormalities in these 2 different platforms. So on top of the concordance, we are able to identify a huge amount of additional information in the form of CNVs, SVs, as well as fusion and translocation in this particular tumor type. The blue arrow shows evidence of chromothypsis of chromosome 8, which is beyond the detection capabilities of SNP microarray, and the bolded genes are the ones which we which are critical or clinically important in the TCGA database, which we use for regular clinical reporting. And as you can see, the comparison of OncoScan data along with optical genome mapping, which highlights the unique things which we have identified by optical genome mapping and the concordance in the black boxes. So here we can look at the complex chromothypsis, which was identified in the chromosome 8 in the optical genome mapping, a large intrachromosomal translocation with MYC, and multiple maps with FGFR1 gene, spanning in the FGFR1 gene. Continuing on the same case, the chromotrypsis on chromosome 8, which is easily visualized in the circus plot, and then both the optical mapping and OncoScan shows aberrations on the chromosome 8, while the optical genome mapping highlight complexities around the chromosome 8, especially many copy number breakpoints with fusion breakpoints, and actionable genes are MYC as well as FGFR1 in that region. Continuing on the same case, we also identified the RB1 deletion on the background of chromosome 13 loss, where there's a complete loss of function of RB1 gene, and the deletion coordinates are very clearly identified along with the size. The next case, this is again a adult brain tumor, female, 43 years old, which shows the concordance for OncoScan and whole genome mapping. The slide shows the CNV profile. The top panel is the OGM data, and the bottom is a SNP microarray, and both the profile shows concordant aneuploidies in 1q deletion, 7, 10, 11, 13, 14, 15, 19, 21, and Y chromosome abnormalities. Again, coming back to the strength of optical genome mapping, on top of the concordance aneuploidies, we definitely have a huge amount of structural variants, CNVs, and fusions. And then, as you can see, the amount of information we can capture on OGM is significantly higher than what we are currently able to do it on a SNP microarray / OncoScan. Here, as you can see, the majority of the concordance is with optical genome mapping with 100% clinically relevant loci. Here we also see in this case the chromothypsis of chromosome 18, and along with additional genomic aberrations seen on chromosome 18, there are multiple SVs which we can see on the chromosome 18. Even though SNP microarrays also shows aberration, the OGM provides better characterization of these aberrations. Here you can see the log ratio along with OGM. Another case, again, this is an adult brain tumor. This is a male, 62 years old. Here, showing the CNV profile by OGM compared to SNP. The top panel is OGM, the bottom panel is SNP, and both the profiles show concordant aneuploidies in 1, 7, 10, and 11. And here, the circus plot shows CNVs and SVs. And again, all the genes which are clinically relevant are highlighted based on TCGA. So not only we are capturing everything we need clinically relevant, but on top of that, we are capturing additional information which has a potential to be clinically significant. Again, 100% concordance with a SNP microarray and OGM in this particular case. And we identified chromosome 10 aneuploidy and partial deletion of P10, which are also called by the SNP. But in OGM, the CNV track on the top shows chromosome 10 loss, and then in the high resolution of nested deletion, which you can see on the copy number profile with approximately a copy number state of 1.2, surrounding copy number state of approximately 0. So you can clearly see the copy number profile in an OGM as compared to a SNP microarray at a gene level. Moving on to the next case, again, this is an adult brain tumors, 58-year-old female. Here it shows the comparison between OGM and SNP, OGM on the top, SNP on the bottom. Both the profiles show concordance aneuploidies in 2, 7, 10, 11, 19, 20, and X. Again, I'm repeating the story case after case, but just to bring a point to each and every one that not only we are having a concordance with what we are able to see on the SNP microarrays, but on top of that, there is a significant additional information OGM adds to the knowledge base in these adult brain tumors. So the same thing here. Here we are highlighting one of the region on chromosome 7, which basically shown multiple aberrations on chromosome 7 with multiple structural variants, as you can see here on the circus plot. Even though the SNP microarray has limitations in showing, the OGM actually better characterize these SVs, including MET amplification, and you can see you can go up to 21, 40, and 21 copies in that particular region of MET. And this information at this granular level definitely helps you to make clinical relevant decisions. So then I would like to put together our summary and conclusions, which we have identified in our initial aspect of validation. So bottom line, there is 100% concordance on all these adult brain tumors which we have seen, and the data compared by OGM versus array, especially the SVs and CNVs. In conclusion, the karyotype FISH and chromosome microarrays are recommended tests globally for routine diagnostic tests for diagnosis, as well as prognostication of adult brain tumors. In our study in initial data, we have shown there's 100% concordance of optical genome mapping versus SNP microarrays. But what I think the importance of optical genome mapping is that its potential to identify unique genomic abnormalities and also better characterize the structural variants, which are at a very high resolution and at granular level. I think it's a great tool for discovery of these novel aberrations, especially the structural variants and fusions in adult brain tumors, which I think were limited in the TCGA data because of the platforms which were used. It is a great tool to assess overall genomic instability and abnormalities, especially the prognostication, especially its use in prognostication theranostics. And I think it might have, it has a potential to gauge the therapy response, very similar to TMB in immune therapy. The optical genome mapping in our study detected multiple clinically relevant actionable SVs events which were missed by SNP microarrays. On top of that, the OGM provided better characterization of chemotherapeutic events. Again, that goes into the earlier discussion on looking at overall genomic instabilities and abnormalities at the tumor level or the whole genome level. So in summary, this data set is our path to develop a lab-developed test on OGM for solid tumor application. And this is based on our experience of using SNP microarrays. And then currently that's what we're using. And I think if this journey continues with the bigger data set and the concordance stays at this level, I think we definitely have a chance to adapt optical genome mapping as a true alternative for assessment of adult brain tumors based on the WHO guidelines. And there are, I think, at least three major advantages. One is reduced hands-on time for OGM as compared to conventional methods, especially SNP arrays or karyotype, a faster turnaround time, again, as compared to karyotype, and it is definitely cost-effective per sample base, again, as compared to a FISH karyotype or microarray. So overall, our initial experience with optical genome mapping in solid tumors has been phenomenal. We're continuing this journey, and we're looking for collaborators and additional partners to put together this analysis of optical genome mapping in solid tumors, especially focused on adult brain tumors. I would like to thank Dr. Nikhil Sirchpal, Ashish Mundala, and Dr. Amin Rojani in my lab and in the institute, and by Nano, Dr. Ben Clifford, Dr. Andy Pang, Dr. Alex Hasty, and Dr. Alka Chobe. Thank you.
Hello, everyone. What an amazing session we had. I will clear some of the welcome to our live Q&A session. William, is that echo for everyone? Hey. Thank you once again to all our speakers, and I think we have most of our speakers here. We have one request. I think Eric has to leave early, so we'll probably go ahead and start with a question for him. So, Eric, can you hear us? Yes. Okay. So you discussed the ability of optical genome mapping to detect genomic instability and focusing basically on clinically to see, how they could determine or be in the PARP inhibitor or ATR pathway inhibitors. So how easy is it to determine genomic instability with optical genome mapping compared to other methods? And how realistic do you think it is to use optical genome mapping for that purpose?
Well, I think the ability of optical mapping to characterize genome-wide instability is similar to whole genome sequencing. and maybe even it might be easier because for tumors that we analyze with both techniques, we usually found more events with Bionano, 1.5 times more. So it's very easy to detect some global instability. The limitation is the ability to detect very small events that can be detected with whole genome sequencing because of the distribution of DLE markers. But we found that most of the time, even when the events were really small, like 10 kb or even less, we could still detect them with optical mapping as insertions, even if there were only one or two DLE markers inside the abnormal region. So it's definitely feasible to detect some genomic instability phenotypes. It works very well for our phenotype of replication stress. I don't think I saw any data on, for example, the BRCA phenotype in breast cancers, but I'm pretty sure that it would work very well as well for this kind of signature.
Thank you. Thank you so much, Eric. Because of all the echo, let me, I forgot to introduce, let me introduce Dr. Sven Bocklund, our Vice President of Communications and Scientific Affairs, who will be moderating this live Q&A with me. Sven, the next one for you. We can't hear you, Sven.
No audio?
Now.
Okay,
now
sorry.
It's good. Yeah.
Yeah, I had the same question for Dr. Broach as well. You also mentioned genome instability. Same question for you. Do you believe that this technology could help you determine whether these unstable tumors with HPV integration could be susceptible to PARP inhibitors or drugs like that?
It's not clear at what point the genome instability takes place relative to the presentation of the tumor or the integration of the HPV. It's something for future. It's not even clear if it's ongoing genome instability as opposed to a pre-existing crisis that led to the integration and the genome-wide rearrangements. That's certainly an interesting direction to go. And clearly something that would be explored. For the HPV positive tumors, these were exquisitely sensitive to radiation therapy and other chemotherapy. And it's not clear whether that's related to the genome instability, but it's certainly possible.
Another question we had for you, Dr. Broach, some of the tumor genome circus plots that you showed only had 10 insertions and deletions. How did you filter the data to have so few? How did you filter out common population variants?
Right, these are all, in these cases, we're made sure that we had blood samples from all of the patients as well as the tumor samples. So these are all filtered against the individual's genome. So that clearly limited us to only those that were somatic.
Okay, makes sense. And then it's more of a comment. I mean, you showed these 26 viral copies in one string. That's just an incredible performance to put that alignment together. I think even with Bionano, that is something like that is just not possible with NGS, is it?
Right, well, it's a great tribute to the Bionano technology and also to one of my first-year graduate students who sat there staring at the Bionano data for several days. So.
Yeah. Okay, thank you.
Thank you. Thank you so much, Jim. Very nice. So Adrian, our next question is for you. So you were part of the team that developed mate-pair sequencing to assess the genomic rearrangements in cancer. And you showed 9 circus plots that were generated with optical genome mapping. Can you speak to the differences in the effort to get to that point with mate-pair versus optical genome mapping and how important in your opinion, that is?
Yeah, so the original development of the assay was when I was at Baylor College of Medicine. And that development of the assay took about 3 years in the early days of which enzymes to use. The original one was using BACs, big BACs, and sequencing the ends. And in the end, we developed methods to do that. Illumina eventually made a kit that does it really well, so we stopped doing that. But it remains a fact that when we still do long insert mate pair mapping in the laboratory, but it remains a fact that calling those structural variants is incredibly difficult. And there is a paper in February, the Nature articles came out from the pan-cancer whole genome working group, and they showed sensitivity and specificity of the callers for structural variants is about 25% precision and sensitivity. And that's just due to the difficulty in mapping these small reads. You're still only getting these small reads at big distances. And so I think that's what you see, this ability to have these long reads, you can map across these incredibly variant regions. And when you have, I mean, you saw a few talks today, when you get this chromothripsis and the DNA is essentially shattered and randomly assorted, you can't, mapping small reads, really confidently map that together. Really, the only way is to do that with a single read, be that, you know, on the RNA or the DNA. So I think that it's just, it is a different way of looking at it. And I think the single molecule sequencers were a real step up because they afforded that, but they're quite expensive and low coverage. So I think it just gives us another way to, you know, unravel this complexity that it's just very hard to do with short reads, no matter how you try to do it.
Absolutely. And you actually hit upon a very important point about the bioinformatic capabilities right now available globally to try and decipher structural variants, and there is no consensus and the performance is variable. So I think that also, you know, speaks to how optical genome mapping data is best utilized. Thank you so much. Sven, back to you.
Yeah, a question for David Chantien related to what Alka said about detecting structural variants with a whole genome sequence. So you integrated many different omics technologies in your work, yet these gene fusions that you discussed were missed by Hi-C and missed by whole genome sequence. Can you talk a little bit about where you think Saphyr's unique strengths are compared to all these other methods that are trying to do the same thing?
Yes, thank you, Sven. So thank you for the organization. I forget to mention that it was very important in that symposium. Yeah, for this identification, the optical mapping, in fact, clarified the splice defects or the fusion RNA that we identified and validated. And to be honest, the different pipeline that were applied on the IC datasets and the whole genome sequencing failed to identify the long-range structural variant. And to be honest, it's a bit. I don't understand why this failed, but this occurred, and this is in a way a problem. But still, the optical mapping revealed those alterations, and this was validated in a way in most of cases by the DM-FISH. So this approach, this optical mapping approach, was very useful for us. So. And probably it's not uniquely because of repetitive sequences. So for the IC analysis, it's a bit complicated, and probably there is some align sequences that were badly assigned. But yeah, the optical mapping solved this issue.
Thank you.
Thank you. Thank you so much. So Eric, thank you. I know that that you have to leave, so thank you so much for participating today. Our next question is for Justin. So Justin, Most of your talk addressed the discovery aspect with optical genome mapping data. And, you talked about the OTS2 deletion and, it being an epigenetic modulator. But you also said that it had never been defined in inflammatory breast cancer or any breast cancer. So can you speculate and comment on that? Because the SVs were pretty obvious from the Bionano data.
It's a good question. I think that, for one, what we've looked at and what we've seen in the actual specimens that we ran on the Saphyr was that, number one, it's not in every cell. And so, there's significant amount of heterogeneity there. And that actually leads us away from a hypothesis that this might be, a germline versus somatic, although we're exploring that in a little bit more detail. So, can I absolutely explain why it hasn't been observed? I can at this point. And, certainly it's known to be sort of an unstable region of the genome, just because it does occur in germlines, these C-terminus deletions. And there seems to be a number of other deletions that are intronic only. So we don't really know why it hasn't been looked at, but I think that probably gets back to the vast majority of data that have been done in this area have been done with sort of short read exon sequencing or whole genome sequencing that may have been missed. In fact, I'll say that the FISH probe that we used, we probably only detected about 20 to 30 cells, like single copy loss. So, we still can't say anything about biological function here. We're sure it exists, and we've been able to validate the deletions in the cell lines, which is a much easier sample type to work with. But, really it won't be until we do the mouse model and really able to assess whether this is actually having a function and impact. I will say we've done a few inflammatory breast cancers now in addition as an update to what I presented, and we don't always see it. So we can't necessarily say it's potentially a pathognomonic feature at this point, but we have yet to observe it in any kind of non-inflammatory breast cancer. I'll also say with inflammatory breast cancer, if this was associated with it and perhaps not necessarily a 100% association, it's an incredibly rare tumor type, and it's very difficult to diagnose. And so, there's limited numbers of investigators really working in this area and examining these type of samples. And to be able to get sample quality, oftentimes these resections are part of a series of formalin fixed blocks, which wouldn't be very amenable to a lot of the higher level sequencing technologies we employ. So I think it's an intersection of technological advance, sample availability, and just how frequently it's being studied, although it is an important question.
Thank you. Thank you so much for your answer. Sven?
Yeah, Alka, actually, maybe a question for you. We got quite a few audience questions about formalin fixed samples. Can you clarify how that works with Bionano, or what tissue we need?
So as I had mentioned in my introduction, right now, optical genome mapping is not compatible with formalin fixed tissues or specimens. We need fresh or frozen tissue material, and this is what all our speakers also spoke about today.
Okay, thank you for clarifying that. And then Dr. Coley, are you there? Yes, there you are. Okay, thank you. We often hear that clinicians don't want to discover too many variants, but you mentioned on several of your glioblastoma cases that you had amazing discovery information, found a lot more clinically important genes. Can you speak a little bit about that balance on how Saphyr can add actionable data that you previously didn't access to, but without overwhelming you with data that is not valuable to you?
So thanks, man. I think thank you guys for putting together a phenomenal symposium. I think it's time for solid tumors to be analyzed the way we need to do with the optical genome mapping, precisely for the reason pretty much every speaker have mentioned here. And going back to the question, I don't think there's any real clinician afraid to look at the data. The challenge is we want the kind of data, number one, it's validated, so we know it's a true variant. We don't want to look at artifacts. Number two, we want to make sure that it's a clinically relevant information. You can always have benign variants, and having a variant of unknown significance is a knowledge gap. At this moment, I mean, if you go back and look at maybe 5 or 10 years ago, the NTRK fusions were variant of unknown significance, but now we know that the NTRK fusions are actually clinically targetable. So at this moment, we are at the stage with the newer platforms, deep sequencing, or even with optical genome mapping, where we're gathering a lot of clinically relevant information, number one, which is validated and true. Once we keep on capturing that data and gather as much as information as possible, and we prove that that's truly oncogenic, there will be people in the background working on targeting these aberrations, maybe CNVs, SVs, or fusions. So I disagree with the notion that we are afraid of new data, but I think I agree with you in the sense that we don't want to push all the information in the clinic to the point that it becomes cumbersome for them to analyze and make sense of it if we don't have any targetable information on these different variants at this moment. But if you look at the reason why we changed the diagnosis as well as prognostication of brain tumors, because the data we captured from TCGA in 2015. If that effort would have not made, we still would have continued the previous grade 1 to grade 4 classification of brain tumors, where we know now that not all grade 4s do bad. It's all depends on whether you have the IDH1B19 codeletion.
Thank you, Ravi. Thank you so much. So we are end of our audience questions, and we just had an amazing session today. We saw all different types of work and integration with other technologies. So I would like to wrap up this session with just closing or parting thoughts from each of the speakers. So since we have Ravi, you on right now, let's start with you, and then I'll ask the next speaker to give your parting comments on this session.
So, I mean, we have been working with the whole genome optical mapping for a while now. I think I get this question asked a lot, whether it is really truly useful in clinical setting. And I think now I'm beginning to answer that question in the sense that I think it's yes or not. It's not a yes and no answer. It's definitely a yes answer. The question is, how are we going to get it in the clinical diagnostics? There need to be, we need to work with Bionano to improve the bioinformatics, lock the bioinformatics the same way we have with next generation sequencing and array systems. And I think once these two things go in parallel, you're definitely going to see the utility of whole genome mapping or optical genome mapping in clinical service. I mean, we're talking about prenatal heme, I think, is a very low-hanging fruit, which needs to be adopted quickly. But I'm a big fan of solid tumors because there's so little done on solid tumors. I think a huge push in discovery for solid tumors, and then using what we have captured in that discovery and then move from discovery to clinical diagnostics. Thank you.
Thank you, Ravi. Thank you, Ravi. Jim?
So I think for years and years, clinicians have had karyotyping available for liquid tumors to be able to get a genome-wide view of the structural variations that are present in those tumors, and that's been remarkable in its both prognostic and diagnostic effects. I think that the Bionano now offers an opportunity to bring that same clarity to solid tumors that hasn't been readily available before. And so I think, as some of the other speakers have pointed out, we're going to have new opportunities to be able to identify new diagnostic and potentially therapeutic tools based on this much more global view of the genome of the tumors.
Thank you so much, Jim. Thank you. Adrian?
Structural variants remain one of the dark matters of cancer genomics. Yeah, I mean, we've had such a focus on the point mutations, and we've learned an incredible amount from the point mutations, but that's because they are easier to detect. That's why there's been such a focus on them, not because they're tremendously more important, but because we can detect them. And the structural variants, many of them remain much of an enigma, and we know there are therapeutics in there. We know there are good diagnostics in there. It's finding the tools to try and extract all that information. And I'm a firm believer that getting the tools out there, getting the bioinformatics nailed down, that we're going to find some really interesting findings from the optical mapping data.
Thank you, Adrian. Really appreciate it. David?
For us and from my point of view, in fact, this tool is highly promising, and I think we should probably improve some part of the analysis, just to find a way to combine the short read sequencing, because this is sometimes mandatory to have the two sides of the coins. But yes, there is a lot of things to do with this technique, and I think we have a lot to discover next, on regulation part of the genome.
Thank you, David. Thank you so much. And Justin.
The other speakers said, and especially Adrian, since it's so sort of close to kind of what we look at, not just from a biology perspective, but also from new targets and things. And one of the ways I obviously from my talk, like, that I think about these kind of technologies is taking these cases with a unique clinical presentation or tumor phenotype in solid tumors, where we sort of look for these what we call pathognomonic features, which are highly related to that unique phenotype, and they often hint at how to better treat these tumors in the clinic. And so I think there's a lot of opportunity for those diseases in which there should be a pathognomonic feature, right? There should be something in the DNA encoding in that tumor, something that causes that unique phenotype. And so in cases where we have yet to, or we've exhausted, whole genome sequencing, whole exome sequencing, other sequencing or other types of techniques and technologies, and still have not found that, but I think it offers, a really unique opportunity to try to find maybe what we've missed.
Thank you. Very much appreciate everybody's comments. Thank you once again for staying till the end. I think that you all have shared a tremendous amount of data associated with different types of solid tumors. And we see the entire paradigm from research to, semi-clinical to really trying to get into a clinical application, along with some basic research work also being done. So thank you once again. Looking forward again to seeing more of your data and expansions on this work in our next session, next time when we have this. But for all the viewers, thank you once again. And I will see you again bright and early tomorrow morning for our next session on complex disorders and repeat expansions. Thank you once again. Thank you very much. Bye-bye.
So that wraps up the symposium today. There's going to be another one tomorrow, of course, and I will be holding another live session regarding that. I just want to make sure that you guys can hear me, so let me know down in the chat section if you guys can. Also, we have about 8 likes left before we start our live session and Q&A, so run up them likes to 150 and we'll get this started. I'll be waiting, and I'll pull up my Thinkorswim app because I know you guys can get there, but it might take you a minute. 150 likes and we'll start. 7 to go. Three to go. Come on, you guys got this. All right, there we are. So we just hit 150 likes. So now the Q&A from me starts, and I just want to say that I'm not a scientist, so if you have any science related questions, just leave that to MKC. She's probably one of the most knowledgeable when it comes to at least this chat. But if you are any sort of medical professional, just leave any like advice or like what you think of these symposiums in the chat section. So I'm going to start out with the price analysis and wait on some questions in the chat. So looking at what's happening today, they're down 4%, 3.7%. They're at 516 currently. And if we look back, I think they found a support right around here. So this is a new support that they created after hours on January 8th at around 4:45 p.m. So it seems like they're bouncing off of that, and now that the symposium is over, it's really hard to tell how people might interpret this and then see if it's going to go up or go down. But what I'm expecting, at least today, if the momentum continues down, we could see it fall down to around $4.91. That's my current support level that I have drawn. But if momentum reverses, if we see a reversal of their pattern today, the MACD is in bearish, but if we see a reversal of that, they could go as high as around 546, I'd imagine. Of course, there could be some bigger catalyst that's coming about, and what that is, we really can't tell yet. A lot of people have been speculating that ARK Invest is going to be buying, but that is, I can't confirm nor deny that. And if you want to confirm or deny that, I will have the link to it on what they buy and sell each day, and I'll put that in the chat for you guys. They send out an email every single day on what they buy, what they sell. You can find that in the pinned comment of this chat. Yeah, there is about a 1 minute delay. All right. So if you guys have any questions about the stock itself, I'm going to start taking questions now. I know that somebody mentioned the option chain opening today. That was Socre stock, so shout out to Socre for pointing that out to me. They did open the option chain. And if we look at it on Thinkorswim, I know it's not going to be the same across different platforms, but on Thinkorswim, we can look at the current strikes. They have a 4, 5, 6, and 7. So there's not a wide range of strike prices yet, but I'm sure they'll start to get added as time goes by and once their stock actually starts to move more. So right now, looking at the call side, the cheapest call you can buy is at the $7 strike, and the bid price for that is $9. So the average price between the bid and the ask should be $95 if you're buying this call option. Does anyone know the status of their FDA application? Bruce, last I checked, at least in their prospectus, and I might be wrong here, so correct me if I am, but last I checked, they said that FDA approval is very time consuming and it's very costly. So, and also from what I read, again, correct me if I'm wrong, but also what I read is if they actually do go through with trying to get FDA approval, then they might have to halt the sale of Saphyr. So it's kind of going to be a double-edged sword if they have to announce that along with announcing FDA approval. So do keep that in mind whenever you start to see more news coming out on the FDA. Yeah, FDA approval does take a long time. I can't give you a real estimate because it's different based on different technologies and exactly how they work, how much more efficient they are, and it just differs from company to company and also from technology to technology, of course. I sound like Trump? Oh man. If I sound like Trump, oh no. You have a question, is anyone here in JAGX? I've heard a lot about JAGX over the past few days, but I haven't done any particular research into it myself since I've been mostly focused on BNGO and a few other genomic stocks that I'm playing with right now. And if you guys want to know the genomic stock that I bought today, you can go check out my Discord, which I'll have linked in the pinned comment here. Just give me 1 second while I pull that up for you guys, and you guys can see what I bought today. Yeah. So if you guys visit my Discord and if you go to Greg's Trades, you'll be able to see what I bought today. But if we go back to BNGO and the price charts, still heading down. Looks like the MACD is still bearish. The RSI is getting pretty low. So the chances of it going up should be higher, just considering how low the RSI is. Since it's oversold, it's more likely to be bought up. But again, that could just continue to trend down. We could see it trade in this range of around 50 to 30 RSI, which is typically a very bearish sign. You've seen this all the way throughout here in this section, and it's just continued to go down. So that's not a really good look for the stock right now, but of course stock prices move up and down by the minute, so it's always subject to change. It's just a snapshot of what's going on in the market, how people feel about it, and prices are always changing. So if I do BNGO news daily, you'll subscribe. I'm not sure if I'll do it daily, but I definitely will be holding tomorrow's symposium live again, just because you guys seem to really enjoy that. And afterward, maybe on Saturday, Sunday, don't quote me here, because I have a pretty busy schedule. I should be posting a video on BNGO and coming up with a conclusion how I feel about this whole symposium, and I'm gonna get all the science from MKC because she's much more knowledgeable than I am, and of course I'm gonna add my own take on it and run through the financials maybe, and then give you a little price analysis at the end of the week. Am I holding any shares? No. I disclaim that at the beginning of this video. I'm not long or short on the stock. I'm neutral, but overall I do have a general pessimistic take on the stock, just because it is so speculative right now on all of these upcoming bull catalysts, and we're not sure if it's actually going to go through. Of course it could go through, but you also have to take into account that it might not go through. And I'm not saying this is like a flipping a coin, but to some degree it is similar in that way. So the upcoming possible catalysts for this stock would be ARCG possibly buying. And from what I've read from Simon's Twitter thread, he doesn't seem too optimistic about their technology, but seemingly he has changed his mind according to somebody on Webull. Not exactly sure how reliable of a source they are, but apparently on Friday he's going to be having a podcast that he's going to put out. And according to this Webull person, he seems to have changed his mind. I'll try to come up with the links for dates for Bull Catalyst, but really there's no telling on when FDA approval is going to come through. There's no telling on when ARC is going to buy. That's completely speculative. It could be tomorrow, it could be next week, it could be next month, next year. We really just don't know yet. And the same thing goes for FDA. Okay. Thank you for linking that, MKC. I will pull that up right now for you guys. So, if we file through this SEC filing and Command F, we're going to look for food. I can't find FDA. MKC, can you give me a page number for where they mentioned FDA? There's going to be a 1 minute lag here, but hopefully you can get that to me as soon as possible. FDA is Food and Drug Administration, and the adoption of their technology really depends on if they get FDA approval. Not really the adoption, but like mass adoption in order to put into clinics, in order to put into clinical research space. Okay, thanks MKC. Awesome. Meanwhile, while we wait for MKC to pull up the page number on that, let's continue to look at the price chart, see where the stock could go. So we're still in bearish territory, MACD. We're still below 50 RSI, so possibly we could see this continuing to go down at least today. But of course, we could always see a reversal, maybe if they make some sort of tweet that people will think is bullish, possibly. Also, I want to thank you guys for attending today. There's a ton of people in the chat, and I really appreciate all of your guys' support of the channel lately. So I just want to thank you guys for that. Public offering, you want to talk about the public offering? We can do that. The public offering was sold yesterday. It was, I forget how many shares it was, but they were for $3.05. So about $2-3 below market value currently. And that was offered by an underwriting. And then they also underwrote some options, I think, I forget again how many shares, but those are going to expire next month, I believe, 30 days. I think is the expiration for those contracts. Sorry if that was loud. I have a very loud and obnoxious keyboard. Probably will continue to fall today until Friday, and that would be the bounce between 5 to 475. Then bounce up on Friday like crazy. And the news continue to be positive. Yeah, so it really depends on if the news is positive or negative, and how people feel about this company and how they're going about the news. And if we look, I think they're probably going to be trading around $4.91 tomorrow, around open maybe. But of course, I don't have a crystal ball here. I wish I could see the future. If I could, then I'd be rich. But let's see. What is Europe FDA approval for? What kind of diagnoses? I'm not sure on that one, Barney. What is the quickest time BNGO could get FDA approval? It's speculative on when you think it might happen. It takes a long time. But as of right now, I'm not sure if they have applied for it. I think they mentioned it in one of their webinars or something like that, but I might be wrong. What are my thoughts on MARA? I haven't done much research on MARA, so I'm not really sure what to tell you on that one. Yeah, I know. There's a ton of pages in that prospectus, so it might take a while for us to find that. But hopefully we can sort that out for you and just try to clarify what they said about FDA approval and what it could mean for the company. Yeah, we'll talk about it tomorrow if we can't find it today. Thanks, MKC. So yes, yesterday they did mention FDA approval in their Q&A session that was held in their presentation, and they're basically saying, from what I'm getting from it, they're saying that they need FDA approval in order for this to get more widely adopted by clinicians, by clinical researchers, and the likes of that. I think they meant FDA, not FDT, MKC. Unless I'm interpreting it wrong. That could be right. All right. So, do you guys have any other questions for me? If you have any other questions, leave them in the chat. Will JAGX follow the same downward trend? I'm really not sure what JAGX is all about. Let me pull them up on Robinhood and see what their company is all about. So, this is commercial stage pharmaceutical company. So they're in pharma, which is separate from genomics or different fields of medicine. But maybe if we look at the charts, we can see some correlation between the two stocks. If we look into, what was it again? JAGX. Okay, I think MKC found it on the FDA approval. So JAGX seems to be in a bullish momentum right now, at least according to this 5-day, 5-minute chart. We can take a step back, look at maybe the 20-day, 1-hour chart. I'm not sure when volume started to pick up. It looks like right around here on December 23rd. I'm not sure what the catalyst was. Again, I haven't done enough research on the stock, but from what I'm seeing right now, we see a trend line that was started somewhere around here. We can draw that up just to give you an idea of how it could continue to move if it does continue to follow this bullish pattern that it's on right now. So we can extend that to the right, and right now they're trending toward the bottom of that trend line. So if we see it break through, we could see it fall down, or we could see it bounce off and then go higher. Not exactly sure on how high it could go. Again, I don't have a crystal ball, I wish I did. It'd be a lot richer if I did, but it is what it is. Page 16, MKC says. Okay. Thank you, MKC, for that. Alright. Page 16. Let's go down, 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 down. Let's see what's on this page. Okay, so here we have what they said about the FDA. I do want to check the date on this just real quick, so let me scroll back up. So this was from what day? They have to have a date on here somewhere. So this was filed on June 28th, 2018, and what they said about the FDA reads as such. Give me 1 second while I scroll down and back to find it. In the future, if in the future we choose to commercialize our products for clinical diagnostic use, we will require to comply with the FDA's pre-market review and post-market control requirements for IVDs as may be applicable. Complying with FDA PMA and or 501(k) clearance may be expensive, time-consuming, and subject us to significant or unanticipated delays. So I think something was written about this in their most recent prospectus, where they said that if they go for FDA approval, they might have to stop sales of the Saphyr for that time being. So you do have to keep that in mind when you start to see more news coming out about the FDA and how that might actually sort of negatively affect their stock if they do also announce that they have to halt sales for some time. Yeah, 23andMe is pretty sure 23andMe is just like your ancestry. I might be wrong on that. Correct me if I'm wrong. Yeah, there's definitely a downtrend going on with BNGO, but again, they could be finding a support somewhere around $4.91. It really depends on how the market feels about their spinal symposium. Also, something that I wanted to bring up is institutional ownership. Again, I think somebody in the chat, I forget who it was exactly, but I think that somebody in our chat said that there was more institutional buys. So I wanted to check that out just to see what's going on in that regard. BNGO, Insider Holdings. And the best source for this information, I think, is NASDAQ.com or Fintel. I'm not sure which one it is. Actually, no. So it's definitely Fintel that has the better information on this. So, here we have it. Institutional shares make up 10% of the company almost. And I think the other day, this number was 5.6, so it does seem like institutional ownership is starting to grow a little bit. 14.7 million shares held by institutions right now. I don't remember the number from last time, but I can go back and I'll verify that with you guys. But if we look here, we can actually see when the institutions bought and see which institution it was, of course. And so the latest filing for an institutional buy of the stock was from SIO Capital Management LLC, and they bought 1.2 million shares, or 1.21 million shares, and they're about a 5%. I'm assuming that means they own that's about 5% of their company. I'm not exactly sure on that though. It's either that or they own 5% of the company. Here's more institutional buys that you can view, and hopefully you guys can actually see this. So this should be all the institutional buys, sells, I think, are in red, I would assume. And then these here, if we go to NASDAQ.com, they might have this better laid out for us. No, so I don't know why, but NASDAQ.com doesn't have the best information on this. Yeah, basically everybody is waiting for ARK Invest, and if they decide to buy in, then the stock is likely going to rally. But of course, again, we don't have confirmation on ARK buying or not, and we're still waiting for Friday, whenever the podcast from Simon Barnett comes out, and supposedly he's changed his mind, but we'll see his new take after the symposium is finished on Thursday. And I think that he might have recorded it previously, maybe yesterday, I don't remember exactly, but somebody from Webull, again, not sure how reliable they are, somebody from Webull said that they recorded a podcast and he seems more optimistic about BNGO's technology. The most recent institutional buy was a year ago? I don't think so. I thought it said 2021, but I might be mixing up my years, so it is the new year. So I get a little bit confused when it comes to that. Let's check one more time just to verify that. We could also check Yahoo Finance. They might be able to tell us. No. Can I share the resources? Sure thing. I will link them in the chat for you guys. If we go to NASDAQ.com, and then I pinned that comment just for you guys. But if we look at the buy date, it was, yeah, Vanguard is the top holder with how many shares? 5.2 million shares. Actually, if we sort by this, so they have 5.2 million shares. Your next biggest holder is Perkins Capital Management with 470,000. It's still not sorted by shares held. Okay, so Vanguard Group holds 5.6 million. Bank of Julius Baer owns 850,000. Geode Capital Management owns 616,000. You can just read through this to see which company holds the most of this. And there's actually seems to be some people shorting it. No, actually, there's no institution shorting it right now. I think that would be negative if it showed the shares held as negative. JP Morgan does own some as well. I can press Control F and see how many they own exactly. So they own 67.3 thousand shares. But also, whenever it comes to these institutions, you also have to keep in mind how big their entire portfolio is. So they might own 67,000 shares, which might be a lot to people like you and me, but in reality, that's kind of the equivalent of retail investors like you and me, again, buying like a few dollars of the stock. So this probably makes up less than 1% of their entire portfolio. And I can't verify that right now, but from what we know, let's see, they have $361,000 worth of BNGO. Vanguard Group has 29 million, almost 30 million in the stock. So you also have to take into account how much, how big these institutions are. If you guys have any other questions about the stock, MKC left the chat, so she can't answer any science questions right now. But if you have any questions about the stock and where it might go, or anything regarding SEC filings, I got the answers. What's my price target for the end of the week? I mean, I can't really give you what price target I think it could go. I think that it could end anywhere between this bottom price level that I have at 424 and then this top price level at 591. So somewhere in this range. I do think that it's going to continue to go down on the symposium tomorrow, just considering how it's moved the past couple of days. But if they announce maybe something exciting, we could see a reversal of that same trend. And if we look closer, I think it's been trending for about 10 days now. And if we look at when the volume started picking up, also something I wanted to mention is that they did, in fact, get cleared of the regulation where they had to stay above $1 for 10 consecutive days. So that means that BNGO won't end up getting delisted. So that is a good sign for the company, but I think that that's also sort of priced into it. So you might see a lot of news articles coming out and saying that, oh, BNGO has been cleared of this, when in reality we probably should have known that yesterday or the day before, considering they were at $5, and the chances of them actually falling by 80% all the way to $1 seem pretty unlikely at that point. But that's just my opinion on it. If we look at the 2-day 5-minute chart, that might give us a better look at where the stock is heading right now. So it does still look to be heading down according to the MACD. They're in a bearish MACD cycle. This could always turn around though. And if we zoom in, we can actually see there was recently a buy signal from the MACD whenever the blue line crossed the yellow line here. Let me get the MacBook, the little dock out of the way. But basically we got a buy signal here. It's heading back up, and we could see a reverse, or we could see it continue to go down. We could see the MACD cross the other direction with the blue line crossing down to the yellow. That would obviously be a sell signal, the opposite to this right here. So just keep an eye out for BNGO stock by the end of the day. My thoughts on it, I think that it could continue to go down maybe to about halfway between this low and this 491 price level, or towards the upside I could see it maybe going to around 526 or so. I don't think there's going to be a lot of price movement, just because the volume is slowing down quite significantly from what they were experiencing a couple weeks ago. But if there's some new catalyst that comes out that could excite a lot of investors into buying into it, and then we could see a reversal. Read the information above. Sure thing, that's me. ARK Invest is mostly in PacBio. September ARK was buying PacBio like every day. Now after BNGO event started, ARK is only selling PacB. So the issue with trying to jump to the conclusion of PacB selling equals BNGO buying is that they're two completely different companies with two different technologies. And also the fact that ARK Invest buys and sells stock pretty much every day, and they're subject to keeping their holdings below 10%. So I'm not exactly sure how much PacB makes up their ARKG portfolio, but we can actually look into that just to give you an idea. I know that it's their top holding in ARKG, but we can verify what percentage. through their website, ARK Invest, and we'll find their genomics page, investment solutions, and I'll also link this for you guys to see. And that's going to be Genomic Revolution, and we can see their institutional holdings by exploring the ETF, and I will send this in the chat. What are the chances of FDA approval? I really can't tell you that because it's based on like different technologies and how efficient they are. We do know that ARKG is more efficient than PacBio's solution to genome mapping, but we don't know the viability whenever it comes to widespread adoption for clinical uses, as right now at least. And again, I just want to say that science is always evolving, that can always change, but this is just what we know right now at least. And if we look at their holdings, Pacific Bio makes up 6.5% of their portfolio for ARKG. Teladoc makes up 6.5% as well. So typically they're selling whenever they hit 10%, but I still can't make the comparison of just because they're selling PacB means they're buying into BNGO, because this company has plenty of money to work with. So I'm not sure why they would have to sell out in order to buy something new. Yeah, that's another thing you have to keep in mind between PacBio and BNGO. PacBio they have two completely different technologies, and PacBio actually has multiple technologies in the genome space, whereas BNGO only has Saphyr. So whenever you're making the comparison, you have to keep that in mind. I do think that there's going to be overall growth in the genomic space because I do think that's going to be the next wave, and BNGO could continue up because of that, but I don't think that they'll reach the value of BNGO. I don't think that you can compare the two values just because of how their businesses differ. Invitae is teaming up with PacBio. I'm not sure on that one, Julio. Or Julio. Sorry if I pronounced that incorrectly. That's alright, I rarely use Twitter too. So, I'm getting ready for lunch, but if you guys have any more questions, leave them in the chat. We can look at the price charts again while we wait for people to leave their questions. And shoot away, guys. I think MKC might have a better grasp on what you're talking about, Asmir. I'm not too great with the science of it, but she's pretty knowledgeable in that space, so she could probably explain how PacB is helping out Invitae. What's my 3-month price target? Honestly, I couldn't tell you. 3 months out, if we look at it, if we look at maybe their 6-month chart or their 1-year chart, it's really just hard to tell where the stock could go just because of the insane rally that we've seen just over the past couple of weeks. And really, it could go either way. It could go up, it could go down. But if we look at the price charts, volume is starting to die off. And if we see something similar to this kind of happened back in July or so, where they jumped up, they kind of fell flat, trended down, and then we saw this exact same thing happen. So we might see it consolidate for a while, maybe trend down, but if we have some good news catalysts, of course, it could continue to go up. How do you buy a call option for BNGO? It depends on which brokerage you're using, but I'll do this on Robinhood, just because I think that's the most used one. But Tom, if you don't use Robinhood, it's kind of similar in this way. But basically, you'll search for BNGO. And then you'll open their option chain, which is to the right when it comes to Robinhood. And then you'll make sure you have buy call option selected. Select whatever expiration date you want. Right now they only have monthlies, and I hope they add more to the chain, but right now this is all they have. So if you wanted to buy one, you would just click on whatever you want to buy. I'm not going to give you a strike price or a date to buy, because I really couldn't tell you. How well will they do on earnings? I think with their software as a service, their earnings is going to improve, but we also have to keep in mind the cash burn of the company. This is something that Tom Nash alluded to, in that they're burning through a whole lot of cash and they're spending a whole lot of money on their executive pay. So that is kind of an issue whenever it comes to their company, and they're spending, of course, a lot more money on research and development as well. And if they want to make major breakthroughs with their technology, they're going to have to pour more money into R&D. But luckily for them, they did do that cash offering, so now they have more cash on hand in order to make those improvements and try to improve their profitability, hopefully. No problem, Tom. What do I think of today's symposium? Honestly, most of it went over my head. I don't really understand the science behind it. But the Q&A session at the end, there was something said, and I don't remember what it was exactly, but it was very interesting the way that they kind of cater some of their questions, at least, to investors rather than scientists. And this is something that I'll probably explain more in detail, but the questions, they kind of try to reword the questions again and again and again to try to get the right answer that people, or the majority of people watching, would understand. All right. Is there any more questions? I'll take one more question. Yes, so I think actually the NASDAQ compliance period ended yesterday, but don't quote me on that. It's either yesterday or it's today. And the chances of them falling to $1, if it is today, is very low, considering that would be a fall of like 80%. I wouldn't imagine, considering the volume right now, I wouldn't imagine that that's actually going to go through. So they are probably going to be NASDAQ compliant. They're going to hold that $1 price level for 10 consecutive days, and they don't run the risk of being delisted. Yeah, I agree. I think that BNGO should be more focused on the researchers than trying to get in touch with their investors. And that's something that I've noticed is kind of a problem with this company. I have my gripes when it comes to that. But again, I do think that, of course, their technology works. It's proven to work, but we need widespread adoption. We need that FDA approval in order for the bull case to play out here. All right, so that's all the questions I'm taking today. I want to thank you guys. Thank you everybody for tuning in to today's live session. I'm going to be holding another one tomorrow. Thursday. That's going to be at 10 a.m. Eastern Standard Time. Hope to see you there. And as always, remember to stay positive, stay green. I'll catch you in the next one. Bye, guys.